think for next year, I kind of want every slide to look standardized. So I might propose using the same, like for every conference TA, use the same.
Oh, like
theme, and then add what you want as your own touch.
Mm. What are some TAs like working on another like format?
Right. I think they can, but
I think like exam reviews, like that could be that like as in like every exam review has this formatting.
Yeah, I think that.
You can put the content that you want, but it has to be in this formatting. I think that would be the happy medium for everything. I'll just give it 2 more minutes. Everything gets situated. Yeah, no, because I'm gonna not necessarily blaze through my slides, but mine are pretty simple.
Yeah, I mean,
Yeah.
there's not a lot of stuff compared to last time.
Yeah.
Even though there's one less lecture,
Just start scaring everybody. This final is cumulative. Everything you learn, you will be applying.
Start again from like oh, it's unfolding everything.
It is?
Alright,
Yeah. Oh, and water too is going to be good. Coffee's good. Yeah, but I'm only going to take something of it. Okay. I'll get started. All right. I guess we'll get started. All right. Welcome, everyone, to the final exam review. I'm going to try, just for a preface, for some of you who may be taking the makeup final or the 5th exam, whatever, just to make up for one of them, I may try to do like a rapid review of every single lecture for those individuals, but that's just a little reminder. But your exam is going to be on this Wednesday from 6 to 7:20. The locations are listed here. I just copied and pasted them into from Canvas onto here, so just make sure that you know where you need to be. And the biggest thing is like only bring a pencil, not a calculator. Like 3rd exam, like people were still bringing out calculators, which is like, it's been 3 exams, but that's all the announcements I have to give so far. The Canvas page also has what's it called? Canvas page also has updated another exam reviews, Q&As, and just be on the lookout if you have more questions and whatnot. In terms of agenda today, we're going to go through obviously all the lectures, but one thing I wanted to mention is that throughout this review session, like you guys have seen in the previous slides, there's going to be practice problems, study tips, whatnot. And what you'll realize is that this exam, as I've been mentioning, is like it's a lot more chill than exam number 3. I know exam 3 was like all the amino acids, and I know they threw on that question about like the neuron and urea cycle. There's nothing like that on this one. It's going to be much more straightforward, and you'll see that on the exam. And then one thing that we tried our best to do is for anything that relates to eukaryotes, we listed them as pink. And then anything that relates to prokaryotes, we listed them as purple. So just keep that in mind as we go through these slides. It's not necessarily like for certain. We may have gotten things wrong, so always like double check. So moving on to lecture 18, this is a pretty straightforward one, DNA structure, topology, recognition. So some of the basics, as we know, DNA it stores all the info, genetic code as specific sequences. So you can have A, G, T, and C for DNA, and then U is for RNA, uracil. And we know that there are 2 types of nucleobases. There's the purines, there's the pyrimidines. Purines are the ones that have 2 rings attached to them. Pyrimidines are the ones that have a singular ring. And DNA is built up of monomers called nucleotides, and multiple nucleotides form phosphodiester bonds to make those DNA strands that we know. So that's just like the very basics. In terms of DNA, RNA, nucleotide structure, I put this slide to emphasize it more, is that you will probably be given like some of these nucleotides, right? And you're going to have to distinguish whether or not it's a DNA versus RNA. And then if they ask you for a nucleoside specifically, you will have to put the proper nucleoside name. So if we're doing deoxyadenosine, that would be the proper name. We wouldn't put deoxyadenylate because that implies that there's phosphate groups on it, but we're asking for the nucleoside. So just keep that in mind, and just keep in mind also of the different sugars. That's pretty much what I'm emphasizing with this slide, is that you'll recognize it, not handwrite it. So moving on to DNA base pairing, just to go over nomenclature stuff, anti-parallel just means that they're parallel, but they go in opposite directions. And then we know that the nucleobases are on the inside, phosphates are on the outside. The base pairing is AT, GC, 2 and 3 hydrogen bonds respectively. I'm going through this really fast because this should be a rapid review, and we'll spend more time on the more complicated stuff like supercoiling. And another thing is the major groove and minor groove. For the major groove, we know that it's wide and shallow, as demonstrated by this orange box, and it allows for and the reason why I'm pointing out the major groove is because it allows for discrimination of AT, TA, GC, and CG. So like the ordering and also the pairing that you have. So in this diagram right here, they have specific colors for the dots indicating whether it's a hydrogen donor, hydrogen acceptor. it wouldn't be a bad idea, especially if you guys are biochem majors and also for MCAT as well, to start memorizing kind of like the specific pattern going from left to right for all of these, because they could ask you on the MCAT. It was asked on mine where they were like, tell me the specific pairing in which order. And so with the major groove, you can distinguish between the 4, whereas the minor groove, you can only distinguish if it's either AT or if it's GC, but you can't distinguish the order between the two. And then minor groove on this diagram is just the narrow and deep. Obviously, the nucleobases aren't as exposed to anything, so you wouldn't necessarily find DNA binding proteins in this region. You would find them mostly in the major groove, because that's where the DNA nucleobases are exposed. Does anyone have questions about anything so far? Okay. So the different types of DNA duplexes, you also need to know this as well. Sorry for this, it's in the way. Let me move this. So there's the A form, there's also the B form and Z. We know that B is the most common, and something that you guys should definitely memorize is like the 10.5 base pairs per turn and the 11. Like, just memorize those two numbers. Know that the A form is right-handed and it's much less common than the B, and that's because for the A form, as you can see here, there's more base pairs per turn, there's better hydrogen bonding, but there's too much of it, if that makes sense. There's too much interaction, and so it causes more of this base pairs per turn. And so that's why you see this increased number rather than the decrease. And then where you would see the A form is the DNA RNA duplexes. So for example, if you have your reverse transcriptase, right, where you form your RNA DNA pairs, that's where you'll probably find the A form rather than B. So that could even be a test question even, just trying to predict. And then there's RNA duplexes, where sometimes RNA, even though we canonically know it as a single-stranded, it can sometimes base pair with another RNA. So that's something to keep in mind. If you ever see like a hypothetical question, you can assume it's the A form and that you know it's 11 base pairs per turn, assuming there's no underwinding or overwinding. And then Z is very rare. You probably won't be tested on it, but it's just good to know that it's even a form in the first place, and the only thing is that it's left-handed. Okay, so in terms of DNA binding proteins, remember the DNA binding proteins bind onto the major groove. So specifically this region where the nucleobases, the A, the T, the C, and the G are exposed. They wouldn't bind onto the minor groove because here you just have the phosphate backbone being like the majority there, and it's occupying that space. So there's no room for those DNA binding proteins to bind onto it. So some common DNA binding motifs you guys should have like in the back of your mind always is these 3, the leucine zippers, the zinc figures, and helix turn helix. And not that you need to know the structure, but more so that you could be asked a test question like, which of the following is not a common DNA binding motif, right? And they'll list literally these 3, and then they'll put like a beta barrel. But we know beta barrel is a secondary structure of proteins. That doesn't mean that they bind onto the DNA itself. So just know that these ones are the ones that bind onto DNA. Anything else doesn't bind onto it. And then all 3 of these contain DNA binding sites, and it's just essentially a sequence of amino acids that bind onto the DNA, that recognize the sequence of the DNA itself. And it's illustrated right here with these 2, and there's glutamine and asparagine because they can form these hydrogen bonds with the bases. You don't need to know all the specific details, you just need to know what I'm trying to get at with this picture is that when these DNA binding sites come in and they bind onto the DNA, they're binding through these hydrogen bonds with the DNA itself. And so just kind of broadening the scope, there are 2 general types. There's sequence-specific, aka they bind to the bases, and there's non-sequence-specific, meaning that they bind onto the phosphate backbone. And if you guys, for your own interest, you can look up polyamines. Those are ones that bind onto the backbone itself. And so now switching gears, moving from the basics and spending a little bit more time on like DNA compaction, like actually breaking this down and supercoiling. So going over the easy stuff, we have eukaryotic DNA compaction, where we know that eukaryotes, we have nucleosomes, which are just, we have histone proteins that allow the DNA to wind around it. And what nucleosomes are the length of DNA wrapped around one histone. And so right here, we can see that, for example, one histone, the DNA could wrap around twice, or it could wrap around 3 times. The number that you should memorize is this 146 base pairs, because that's typically the average number of DNA that's wrapped around a histone core. So we don't know how necessarily, this is a cartoon picture, so it could be 3 to 4 times that it wraps around, but we know like for certain that it's 146 base pairs when it loops around the histone protein. And then for the histones themselves, they're small proteins that are wrapped around the DNA, and we know DNA is negatively charged, so it makes sense that these histones are made up of lysine and arginine residues. They're positively charged, and they're going to bind onto the negative and form that very stable electrostatic interaction. So that's a good property to know in your mind, that histone proteins are positively charged, not negatively or neutral. and that causing, for example, I'm throwing some stuff out there, but let's say we mutated a histone protein that mutated the amino acids from positive to like a negative or even a neutral charge. What would you expect to happen to DNA compaction? Well, because it's not positive anymore, you're going to have less compaction because there's not, those electrostatic interactions are not forming. So once you mutate the histone proteins and you get rid of the positive charge, the DNA binds less stronger, binds more loosely. And then talking about the nucleosome core particle, it's composed of two different subunits each, so 8 total. So there's 2 of the H2A, 2 of the H2B, 2 of the H3, and 2 of the H4. There's 8 total that form the histone itself. And then in terms of nucleosomes, histones, all this kind of wrapping around, it's usually found in chromatin of all eukaryotic cells, and it allows us to be able to package the DNA much more efficiently on top of the supercoiling. So going into a little bit more in terms of regulating post-translationally or post-modifying it, whatever, of these histone proteins, we know that for this first part, this diagram, we know that the tails have lysine residues on them, as I mentioned, they're positively charged, and these lysine residues can be acetylated. And what that means is we go from positive to neutral. And when this happens, we lose that positive charge, and it's not going to bind onto the DNA. The end result is that the DNA is bound more loosely. And what this correlates to in terms of chromatin organization is you now go from heterochromatin, very tightly packed, to euchromatin, where the DNA is loosely packed, and the DNA itself, those genes can become expressible, if that makes sense. So acetylation does that, where we go from heterochromatin to euchromatin, and then deacetylation is the opposite direction, where we go from euchromatin to heterochromatin. And this is another picture showing what the euchromatin and heterochromatin is. Something to keep in mind as well to make sure you're straight on all that stuff. And then this is just a depiction of how this compaction could form these chromosomes, essentially. Does anyone have questions so far about like regulation of histones or identity of histones and whatnot? Okay. And then this was something new in your guys' class. It wasn't presented in mine, or I don't even think last year's, but the long non-coding RNAs, which are essentially just these, you can think, well, I'm not going to confuse, but anyways, the RNAs themselves, these are RNAs, right? This like green loop, and what ends up happening is they can bind onto the DNA and form like matched sequences in a way. It's almost like microRNAs, where they kind of they form the complementary sequences to the nucleotide sequence that we're looking for. And so in the similar way here, the RNA kind of forms these very favorable interactions, and then from there, the RNA is bound to these DNA binding proteins, and essentially it allows for us to like manipulate the DNA to move it around. And so this allows us to compartmentalize using, again, the long non-coding RNAs to compartmentalize the specific types of DNA. And this is just something new, so like if I probably wouldn't imagine it would be emphasized too much on the exam. This would probably be like a very low yield. Okay. So now onto the harder stuff, at least stuff that I found confusing the first time I took this, is DNA supercoiling. And this one is more pronounced in prokaryotes because they don't have the histone proteins, right? For eukaryotes, we do have the histones, so we don't necessarily need to rely on the supercoiling as much. And so just to give a general overview, we have like a relaxed, these are like fake numbers essentially, they could just give you other ones as well on the exam, but we have a relaxed, and they'll tell you specifically, a relaxed chromosome, a circular plasmid, I guess we'll define it. A circular plasmid, it's relaxed, there's 8 turns, and then there's situations where it can become strained, where it underwinds. And the way we see this is through DNA replication, but I won't jump the gun yet on that, it's on the next lecture. But what I want to go through is that in order to compensate and get it from the strained 7 turns back to the relaxed 8, the plasmid can basically coil in on itself and form like this figure 8, and that will put it back to the relaxed state. So that's why supercoiling is useful, is because it alleviates that torsional strain that's caused from this underwinding or overwinding, either or. So in terms of circular plasmids, this is a better picture to show you, but concept I want to bring up is linking number. So as you can see here, this is a linking number of 1, meaning that we form one loop through another like circular plasmid. And so if, for example, if there was a cut in this plasmid, right, on this left one, we wouldn't be able to define the linking number because, again, it's not linked per se. So it has to be a full like closed loop within the other one. And just to make it a little bit easier, on this one, we have a linking number of 6, where we form 6 loops. So there's 1 here, 2, 3, and so on and so forth. There's 6. They would not give you a picture and say, what's the linking number of it? What they would ask you is, this is the number, this is how long your plasmid is, base pairs wise. and this is how many turns it makes, calculate the linking number. That's what all they'll ask you. It's a simple like calculation. So but before I go on, like just don't bring calculators, but you can just even like write out the division. It shouldn't be too difficult. And then in terms of this picture is very useful in terms of finding out or what it means when something is a negative supercoil or when something is a positive supercoil. So in a relaxed DNA, it's just circular, and sometimes when it underwinds, it'll basically form this negative supercoil. When we mean by negative is that the delta LK is negative. That's what we mean by negative supercoil. It's not that it's less or whatever. It's just that we have less turns, I believe. Yes. Hopefully I'm not getting that wrong. And then in terms of positive supercoils, we're just having the delta LK just be a positive value. So as something to keep in mind whenever we talk about positive and negative supercoiling, this will become also very prevalent in topoisomerases, which I think is the next slide. And then something new as well that Dr. Holmes brought up is superhelical density as well, which is just a measurement independent of DNA length. All it is you're taking your change in LK and then dividing it by the original LK value when it's just relaxed. And this will tell you, even though you may not know how big the DNA strand is, you'll be able to isolate in that little part the superhelical density, how much it's turning at that specific point. That's the function of this. I don't think he'll test you conceptually on it. I think he'll just give you like basic calculations. And it'll be very like easy. It won't be anything to trick you or whatnot. Anyone have questions about like this partly supercoiling? I'll talk about how it's related to the topoisomerases in a little bit. Okay. So in terms of topoisomerases, so Holmes did put the mechanisms on here, but just to like simplify it like very easily is that when we're first starting this reaction, what we're gonna do is basically break one strand of DNA. How do we do that? Well, we have to break the 5,3 phosphodiester bond linkage. And that's exactly what this mechanism is going through right here on the left side, where we have a tyrosine residue basically it's gonna break the phosphodiester bond, and that's what ends up happening in the first place. And that's where we get this, where the tyrosine is bound to the 5' end of our broken strand and 3' end is exposed. And then basically the topoisomerase 1 passes one strand through and then ligates it. And then this is just the ligation step right here that's showing. So you don't need to like go into every single specific detail. The only thing you need to know is that in order to cleave it, the amino acid from the topoisomerase is going to come in, act as a nucleophile, and basically break the DNA strand. And then for this one, basically the strand just wants to reform that phosphodiester bond with the DNA, and it's just going to, again, do a nucleophilic attack from the 3' end, and it's going to attach itself to the 5' phosphate and then reseal it, essentially. That's the first mechanism. It's not necessarily like too hard conceptually, but one thing you need to know about this is that there's no ATP hydrolysis with type 1 topoisomerases, whereas type 2, we definitely use ATP. So that's a distinguishment that you guys should definitely have solid when it comes to the exam. And then in terms of type 1, we know that it can either do plus or minus 1 for the delta LK, so it changes in increments of 1 rather than 2 for the type 2 topoisomerases, and it can remove negative supercoils. But now bridging onto the type 2 topoisomerases, because this is one where I want to talk more about, there's two different gates here. There is the N gate and there is the C gate. And an easy way to memorize the gates is like when we label the amino acids, we go from N to C. And in this case, for this mechanism, we go from the N gate to the C when we're transferring and passing the two strands through it. So in this first part of the type 2A topoisomerase, the strand that we're going to cleave, we're going to do this double cleavage, is going to go in the N gate, which is this top part right here. And basically, once the DNA enters, it's going to change the conformation of this topoisomerase, and it's going to close it and basically lock it in place. Once it's locked in place, the topoisomerase is going to undergo ATP hydrolysis to cut the DNA, pass it through into the C gate, and then from there reseal it. And then now you alleviated the underwinding or overwinding, depending on which one. Now, one thing I really want to emphasize that is only in type 2 topoisomerases is that it can introduce negative supercoils in prokaryotes only, not eukaryotes. So that's something to keep in mind. So in terms of this diagram right here, let's say we already have the relaxed DNA, right? If we introduce a type 2 topoisomerase, we said we can introduce negative supercoiling. So we can decrease further by -2, whatever from the original value is. So if, let's say, we're working with these numbers, right? On the exam, Holmes says, you have a relaxed DNA that has an LK of 200. and you subject it to one round, and this is in prokaryotes, you subject it into one round of topoisomerase type 2, like a type 2 topoisomerase. What ends up happening? Well, we know that it doesn't go in the positive supercoiling direction, so the answer is that it'll decrease the linking number by 2. It goes down by 2. And so that's an application that you would just have in terms of testing him testing you on type 2 topoisomerases. But does anyone have questions about these two at all in terms of mechanism? It's not too bad. And these are like one of the very few mechanisms you have to like have memorized per se. But okay. So that was lecture 18. It was much more the basics, but now we'll actually start moving on to DNA replication and repair. So I'm not going to go over like the other two models, but we all should know that there's 3 models. There's conservative, semi-conservative, and dispersive. The one that's correct is the semi-conservative model, where basically our parent strands break into 2, and then the replicated daughter strands are going to be on each one. And so you form this hybrid DNA essentially from the first round of replication. But we won't go into the experiments. That is something you guys will have to know on the MCAT, the Meselson experiment or something that proved that it's semi-conservative. But going on to the basics of DNA replication. So DNA polymerases require 3 things: the template, the template strand, which obviously like that makes sense. If you don't have the template strand, you're not going to copy anything. And the second thing is the RNA primers with a 3' OH. That's in the sugar itself. Then the dNTP substrates, meaning these incoming nucleotides need to come in. And this just makes intuitive sense if you guys have seen replication before. And we know that DNA polymerase catalyzes or has a 5' to 3' synthesis. So to get this like very straight, we have our template strand going from 5' to 3'. Our DNA polymerase is going to read it from 3' to 5', and that's going to synthesize your 5' to 3'. And that still maintains the anti-parallel. So something to always keep in your mind is that whenever it forms the new DNA strand, it's still going to be anti-parallel to your original one. So that's how you know that you're doing it like correctly, is that the 5' to 3' should be anti-parallel to the original parent strand, essentially. And then something about the active site, it's spatially restrictive. All I'm trying to say here is that it mainly accommodates for the correct base pairing, but we obviously know there could be mismatches, but that's very rare. And that's because, again, the active site is spatially restricted, but it's not 100% perfect. And then some key features about the DNA polymerase is that it has the magnesium, and it is coordinated with a highly conserved aspartate residue. And essentially the magnesium is it's a useful cofactor because it will essentially help stabilize this oxyanion that forms when you add these incoming nucleotides to it. And so in the mechanism, we have our 3' that's growing, right? It goes from the 5' to 3' direction on the new strand. The 3' end is going to be nucleophilic. It's going to have that negative charge, that high electron density. And so from there, the incoming nucleotide is going to bind favorably to the complementary base on the template strand. And it's going to form the hydrogen bonds that kind of stabilize it in the active site and get it coordinated. And then from there, the 3' end is going to attach itself to the 5' end and form that phosphodiester bond. Does anyone have questions about that part so far in terms of the mechanism? Okay. And then on to part 2. So DNA polymerases also contain the 3' to 5' exonuclease activity. What that means is, while it's synthesizing from 5' to 3', it's also reading what it just put in the opposite direction in the same enzyme. And so basically it just allows for proofreading of the nucleotides it added in, and this improves the accuracy by 100-fold to 1,000-fold. So it just makes it much more efficient and less subjective to like DNA mismatches. And then something I wanted to note is that was DNA polymerase 3 that I was talking about. DNA polymerase 1 also contains this 3' to 5' exonuclease activity, but it also contains a 5' to 3'. And this is important when it comes to, we know that DNA polymerase 1 is going to be used for a lot of these mismatch repair mechanisms, which we'll go into in depth. And then I want to bring up this concept of processivity, which just means how many consecutive reactions can an enzyme catalyze before it releases its substrates. So essentially, applying it to this material, if the DNA polymerase binds onto the DNA, how long will it stay on before it falls off? How long will the strand become, or at least the daughter strand that it's replicating? And so for DNA polymerase 3, we know this is the main person that's replicating the entire DNA. And it's not necessarily a gene, it's the whole DNA itself. And so it's going to have high processivity. And from the table that you guys had, it was 500,000. You don't need to know the specific number. It's just that it replicates a lot more base pairs than DNA polymerase 1, where it has low processivity. It has from 3 to 200 base pairs. And this makes sense. DNA polymerase 1 is going to be used for the mismatch, whereas DNA polymerase 3 is going to be used for the actual replication itself. So you have questions about that? Cool. And then now going into the basics of DNA replication, as we know, there are 3 stages. There's initiation, elongation, and termination. We know that initiation, we need to unwind the DNA itself, so it's very wound up. We need to basically open it up, have the strand separation, and so we'll go into the details of that. We know elongation, it's not just that DNA polymerase 3 just comes in and starts replicating. We first need to lay down the primers using RNA primase. Then after that, we need to polymerize using DNA polymerase 3, and then we also need to ligate it, the strands that are created by the Okazaki fragments from the lagging strand. We need to connect all those together using a ligase. And then the last one is termination. How do we stop the DNA replication once it's complete? So talking about initiation, there are these DNA unwinding elements. All that means is that it is a region, a specific sequence that has a high composition of AT base pairs. We know AT base pairs have 2 hydrogen bonds rather than GC having 3. When it has the 2 hydrogen bonds, it's much more likely for it to strand separate. And so that's where we're going to find majority of our replication forks. That's where the TM is going to be lowest, because we know TM has to do with the 50% of the DNA denaturing and the 50% being still natured. It's still bound together. And so in terms of that, DNA helicase is going to continue to unwind the DNA once it's found its region in these DUEs. And so it's going to continue unwinding the DNA, basically separating the hydrogen bonds, and it's going to use ATP hydrolysis to do this. So that's something to keep in mind. And DNA gyrase, which is a type 2 topoisomerase, is going to be ahead of this complex, this like protein complex. It's called the replosome. It's going to be ahead of it and basically alleviate that torsional strain. I don't know if you guys have ever had like cables like wound up, like all mixed up, and you try to just go from one end and just basically alleviate it. It sometimes gets really hard when you get to the other end and the cord is just all tangled up. So the DNA gyrase is going to alleviate some of that torsional strain. So it makes it a lot easier to just straighten out the DNA strand. And then lastly, we have these single-stranded DNA binding proteins, where if we separate the DNA strands, they do really want to go back together. So these single-stranded DNA binding proteins are going to bind onto the single strands and just basically form like favorable hydrogen bonding with the single DNA strand to prevent it from collapsing, from going back together. And so this just keeps that replication bubble like continuously like separated as the DNA becomes replicated. And then moving on to elongation, as I mentioned, the replosome contains all the proteins needed for our DNA replication. So in this table, these are all like proteins found within the replosome. So they're connected somehow. It's in the same protein. And so these are just the general functions, but I want to start with in terms of the first step of elongation, which is setting down the RNA primers. So in this bottom left diagram, we see that we start at the replication fork, and we need to set down our primers going from the 5' end to the 3'. So we're synthesizing these RNA primers from 5' to 3', which means that we're reading the template strand, the old strand, from 3' to 5'. We're still maintaining the anti-parallel. So that's just the easiest way to not get confused is the DNA is always anti-parallel, whether it's the newly replicated or whether it's the old one. And then this is just the start of this initiation phase where we have as I mentioned, the DNA gyrase is very much ahead of this replosome. It's just alleviating the torsional strain. And then we have the rest of the stuff doing its thing, the helicase, the primase, the SSBs. Those are also there laying down the primers and holding out, basically keeping the DNA separated for the initiation phase. And then moving on to the second part, which was a little bit confusing the first time I was learning this stuff, which is like which one is the leading strand, which one is the lagging strand. The best way to keep this like not confusing or anything like that is to figure out the direction of the replication fork. And you can just draw like a giant arrow for this. And then you're going to have, and also like label like the strands 5' to 3', and then the opposite direction for the other strand. And essentially, whichever direction that you're going with the replication fork, that's your leading strand. And you just have to make sure that whenever you label the numbers for your newly replicated, that it is anti-parallel to your original strand. So I like in terms of remembering the numbers, I wouldn't even stress about it until you start doing the lagging strands. So with the lagging strands, the replication fork is opening one way, and the lagging strands are synthesizing in the opposite direction of which the replication fork is opening. So right here, as we can see, the replication fork is going from left to right, and then the lagging strand, it always synthesizes from 5' to 3', and this is going from right to left. And so I mentioned this in like one of my videos, but essentially like if you think about it like in 4 quadrants, 2 of the diagonals are going to be the same, either leading or lagging. You have to figure that out, and the other 2 are going to be leading or lagging, the opposite. Yeah, and then that's just everything I need to say. Does anyone have questions about like leading versus lagging strand? I'll go into the importance of like what the machinery looks like from there, but just about this material so far. Yeah.
I don't know the leading or lagging strand, but just a question about DNA gyrase. You said it's ahead of the replica.
Mhm.
Does it relieve overwinding or underwinding ahead of the replica? Or do we not need to know that?
Intuitively, it would alleviate, like, I don't think you really need to know this, it wasn't tested, but intuitively, I think it would alleviate underwinding because you're basically, when you're separating the strands, you're causing more winding at the rest of it, of the DNA, and so I think that's considered underwinding, if I'm not mistaken. But yeah.
A little bit of both. You don't want to keep it, you don't want to lose all your DNA as single strands, but you also don't want to keep it too tight so you keep moving on. But yeah, it's a balance.
Yeah. Any other questions? Okay. So in terms of this is what I want to talk about in the difference between the leading strand and the lagging strand. For the leading, one thing I want to emphasize is that DNA polymerase 3 has these beta clamps that allow it to stay bound to the DNA. So the reason why DNA polymerase 3 has high processivity, why it stays on the DNA the longest, is because of these beta clamps helping. And so in terms of like the leading versus lagging strand, in the leading strand, there's really only one beta clamp because in the leading strand, it's following the replication fork. You only put down your primers one time at the replication fork origin. Whereas on the lagging strand, we're making all these Okazaki fragments, so we need more beta clamps to come in and hold the DNA polymerase 3 in its way. And so right here, I just want to show you is that there's these beta clamps that are left behind, or they get bound to the primase, and then the core, essentially the DNA polymerase 3, is going to come in and basically attach itself to the beta clamp and then allow for DNA replication. So the biggest thing to take away from this slide is that DNA polymerase, the reason why it has high processivity is because of these beta clamps, and you really only need one beta clamp for the leading strand, and then the lagging strand, you just need a lot of them because you form those Okazaki fragments. And each time you have a beta clamp loaded, it needs to have its accompanying RNA primer as well on here. Does anyone have questions about that, the beta clamp? That's really like what I want to emphasize with this slide, not all this like complex stuff in the protein. Okay. And then sometimes during the elongation phase, like we either need to ligate it or we need to have some exonuclease activity. So this yellow enzyme here is now your DNA polymerase 1, where if we're going, I'll skip that. I'll go to ligase first really quick. I should have switched the order. But when we have these fragments, right, where we have these little gaps, we're going to use DNA ligase to come in and essentially fill in the gap here. And so this is the mechanism that Holmes had you guys, like, it was put on his slides. And so the takeaway from this is that with the enzyme DNA ligase right here, it's going to be, adenylated using ATP or AMP, whichever one it is. And it's going to attach itself onto the enzyme. Then from the enzyme, the adenylation is going to attach itself to the 5' end of our nick in the DNA. And so from here, as you can see, this is the 5', and we attached our AMP to it. And then when it's attached, that adds enough energy, because remember, the energy is from the phosphate to phosphate repulsion right here. So the AMP has that phosphate group, and we also have the original phosphate from the DNA strand. This repulsion, when when this repulsion is here, it powers our 5' end of the DNA, and the 3' end of the DNA itself that needs to fill in that gap will basically do nucleophilic attack. So in a way, you can, I was going to say you can think of it like the CoA stuff with the enzyme, like the thioester linkage, how it activates it just a little bit, but I don't want to confuse people. Just know that this is being activated essentially to fill in that gap and to allow that phosphodiester bond to occur. And then in terms of DNA polymerase 1, it has the 5' to 3' exonuclease activity, meaning that it'll literally take off some of the parts of the DNA that's mismatched on the right side, going from 5' to 3'. But additionally, it can also have 3' to 5' exonuclease activity as well. Or sorry, it can also have 5' to 3' polymerase activity, where it will also add in DNA nucleotides as well. And then moving on to the termination, this is for prokaryotes only because, again, this is circular DNA. In the prokaryotes, they have these termination sequences, which just allow the DNA polymerase to just kind of fall off, and then it stops DNA replication. And typically, once we reach the end of DNA replication, as shown on this figure on the right, we end up with a catenated chromosome, which just means it's still intertwined. It's not separated. Like if you were to try to pull it apart, it's still there and bound. And so what we use is a DNA topoisomerase 4, which allows us to separate these two strands. You don't need to know the mechanism. You just need to know that it's a different isoform of the topoisomerase. Instead of it being topoisomerase 2, it's topoisomerase 4. And that's pretty much it for termination for prokaryotes. But for eukaryotes, we know that it's linear. We know that it's not circular. And so the solution the problem that we run into with linear DNA is that when we reach the ends of them, for our lagging strand, we never set the primer in the first place for the Okazaki fragment. And so we're going to have this very little gap where there's no DNA. And we want to fill it because there's important proteins that we need to make from the very tail end of the DNA. So the solution to work around it is using this telomerase enzyme, where it has its own RNA primer bound within it that's going to match complementary to the end sequence of it, and it's going to basically attach itself to the end, elongate the whole DNA, and then allow for us to replicate the sequence that we want and a little bit more to form those telomere caps, essentially. Does that make sense, or was that a little bit confusing, or should I clarify something else?
Also important to know that telomerase is not a permanent solution. Like, that's why we still age, because it's sort of a temporary solution over time. It tries to keep elongating, but when the telomerase leaves at the very last part of the diagram, notice there's still a little bit of a gap. So you can never fully fix the gap. Keep that in mind.
And I also want to mention that telomerase is technically, like, this could be like a matching question or something, but telomerase is a reverse transcriptase, which means that you're taking RNA, or it's essentially using RNA to build up DNA from it, because that's how reverse transcriptases work, is they go from the RNA to the DNA. So something to keep in mind if they ask for like these very like subtle differences or like definitions. Yeah.
So is it filling that little space, or is it just like lengthening it so that?
In order to fill that little space, it has to lengthen it a little bit. Does that make sense? So when it comes here, right, onto this end, the telomerase is going to elongate it even more, a little bit, and then from there, the primase will be able to come in, insert the RNA primer, and then finish replicating.
Yeah.
It's like
Yeah.
you're just adding in like buffer material, basically. Enough buffer so that you can finish the rest of that blue line, which is like important genetic material. The rest that it fills in, right, it sort of has like a buffer, a cushion that you don't really care about, that can be afforded to, like, lessen over time. Yeah.
So do you need to know about those two proteins in the bottom right?
The TRF1 and TRF2? No. Like, you just know that they're telomerase proteins. No. Any other questions? Okay. So switching gears into like DNA mutations and DNA repair. The mutation stuff, there's 4 of them that you guys need to know in terms of transitions, transversions, base insertions, and base deletions. For the first one, for the transitions, we basically keep the same ring, but we change the identity of it. So it's a purine to purine. So it's a 2-ring nucleobase to a 2-ring nucleobase. So we go from AT and we convert the A into the G. And then from there, it's going to base pair with its complement. So that's what a transition is. Whereas a transversion is essentially converting a one form of the ring to the other. So for here, it's pyrimidine to purine, from a 1-ring to a 2. And then on this bottom one right here, we go from a 2-ring purine to a 1-ring pyrimidine. And so that's what a transversion is. So here we go from thymine, which is a pyrimidine, to adenine, which we know is a purine. And then vice versa on this one on the bottom. And then for 3 and 4, they're pretty intuitive. Base insertion, we're inserting a base into the DNA. It basically expands the sequence by 1 nucleotide for insertion, or it shrinks it by 1. And you know that later downstream in translation, it's going to shift the reading frame. And then mutations can be silent, deleterious, or advantageous. Silent meaning that they don't change the amino acid. Deleterious meaning that, or sorry, in this case for gene mutations. Silent means that there's no impact on gene function, which usually corresponds to not changing the amino acid itself, the amino acid profile. Deleterious just means you impair the gene function, so you can have like a missense or nonsense, but That's towards the translation, translation aspect. And there's advantageous, where you actually have a gain of function. You introduce a new function, or you increase the efficiency of it. So those are like the three effects of these mutations. But these are the four that you should be very familiar with, because they could ask like a question of saying like, it's a transversion from thymine into an adenine. Like, tell me specifically what the change is, and you'll just have to identify that's a pyrimidine, to a purine. And then here are some ways that the DNA itself, like biologically, can be changed and mutated. So the first example here is the deamination. And deamination, all it means is that we're removing an amine group. So the best example that you guys should have like memorized per se is the cytosine loses its amine group and becomes uracil. And so you really need to have this bottom one memorized, the methyl, cytosine, and thymine. It's just more so that we're losing the amine group, and it changes the nucleobase profile entirely. So we go from a cytosine to a uracil, and we know uracil isn't found in the DNA. So this is a mutation. And then in terms of these other ones as well, they also occur. I'm just showing you as examples. And then these are the nitrous acid precursors that cause deamination to be more likely. They kind of induce it in a way. But the only thing you really need to take away is that deamination, you're just getting rid of the amine group from the base itself. Yeah?
In the deamination in this example, it also includes adding the carboxyl group?
Yeah, typically for the, for this example, when we talk about deamination DNA, we think of adding a carbonyl group in there in the first place, and not just thinking as like a methyl per se.
Okay.
Yeah. Any other questions? Okay. So then another example is depurination, which depurination, it's kind of in the name. Depurin, so we're getting rid of a purine ring. So you can find this in guanine or adenine, so one of the two bases that are purines. And what ends up happening is you basically break the bond between the nucleobase and the sugar, and you form a apurinic residue. And I'll explain why this name is important on one of the replication mechanisms. I'll emphasize it again. But essentially in depurination, we're getting rid of the nucleobase. So we're essentially removing that base from the sugar itself, and it causes aberrant interactions with the DNA itself, or you lose the hydrogen bonding in the first place. Now, this is the third example, which is alkylation, which we essentially just add an alkyl group, a CH3. And in this example right here, we have cytosine, which gets methylated and becomes thymine. Let me see if I can find it. So yeah, essentially, the guanine. The guanine becomes methylated at the position number 6. And when it becomes methylated, you lose this hydrogen bonding from here. And what ends up happening is that this methylated guanine will only pair to the thymine itself, so not the cytosine. So this is a way in which methylation can get rid of a hydrogen bond and basically make it so that it base pairs more favorably with the thymine rather than the cytosine. And here's some examples of alkylating agents. They probably won't even ask you to recognize it. I'm just putting it here for your information. But this is the biggest thing that you want to take away, is that methylating the guanine at the 6th position of the oxygen. And it's 6 because if you were to number these carbons, it goes from 1, 2, 3, 4, 5, 6 here. I don't know why the numbering is like that. That's just how it is. 6 is here. And then at the 6th position, it gets methylated, and it forms the 2 hydrogen bonds with the thymine rather than the cytosine. Any questions on that? Okay. And then moving on to a more classic example is UV irradiation, where we have 2 adjacent thymines where they're basically base stacking with each other. One's above, one's below, and we're adding UV, which we know has higher frequency, and that means using like Planck's constant, whatever, it has higher energy as well. Because it has higher energy, you can do these like weird radical reactions, which you guys do not have to know, but it'll essentially form this thymine dimer. And it'll basically kind of make these covalent bonds between the 2 adjacent stacked bases. When this happens, it changes the geometric configuration of the DNA itself, and it forms this kink. And that's really all you have to know about this, about the UV irradiation, is that you add a lot of UV light, which has high energy, and it causes a thymine dimer to form. And when this dimer forms with these covalent bonds, it generates this kink in the DNA itself. And that kink is bad because it causes all the nearby bases as well to like change and to not have a stable of hydrogen bonds. And then onto like the actual repair mechanisms themselves. Those are just examples of how biologically DNA could be altered or mutated. So moving on to the repairs themselves. For mismatch repair, the way it works is that there's a certain window of time where the DNA will become methylated, And essentially, that's how we're able to distinguish between the daughter strand and the parent strand, where the daughter strand isn't methylated yet. It will be soon, but it hasn't. And so during this window of opportunity, these MUT like proteins, like you don't need to know like L, S, or H. Just know that it forms a mute complex, MUT, and it's 3 of these proteins that you really need to know. But what ends up happening is when this complex forms, it's going to form like a kink in the DNA, as you can see here, like it's changing the configuration of it. And what ends up happening is this complex will basically cut the newly replicated DNA, not the old, just the new. Because remember, the old hasn't mutated yet. It's the new one that has it has a mismatch base in there. So it's going to cut the new one. When it cuts it, you're going to have this exonuclease activity. The exonuclease activity is going to come in. It's going to be like a DNA polymerase, I think, or helicase, I think, or I don't even really know. It's exonuclease, an enzyme that's going to come in and essentially start taking away the DNA chain, and then from there, once it makes it to the mismatch repair, that's when our DNA polymerase 3 is going to come back in with the SSBs and finish replicating it, which is not shown in the best there. But anyways, on the slides themselves, you'll see that DNA polymerase is finishing up the replication and fixes the mismatch repair. Does anyone have questions about this? Yeah.
When would you prefer like one over the other?
What do you mean, one or the other? They're just showing different directions. It can occur either or, but they wouldn't ask you to distinguish it. You just need to know that it's going to go downstream or upstream of the two. Because again, like it forms this like loop right here, so it can either cut in the right direction, I guess, or in the left. It can pick either or, and that's what this is demonstrating here. Is that this one made the cut on the left side, and then the other one made a cut on the right. But it can be either or. Any other questions about this repair mechanism? Okay. So moving on to the base excision repair. So in terms of this, I highlighted in this pink box right here, we have the uracil. And we're going to use this new enzyme that you guys haven't used before, is DNA glycosylase, which all it is it's going to be an enzyme that basically cuts the bond between the base and the sugar. Hence why it's called a glycosylase. When it cuts it, it's going to essentially create a apurinic residue. Remember how I mentioned in these few slides, you form this apurinic residue where it doesn't have the ring anymore on it. So that's exactly why this is called an AP endonuclease. Apurinic. That's what the initials stand for. And with this AP endonuclease, what's going to end up happening is you're going to cut the phosphodiester bond between the aberrant base and your regular strand. And when it cuts it, you're going to insert a DNA polymerase 1 because we know it has that 5' to 3' exonuclease activity on top of it also incorporating the DNA itself. It basically does both features in one. And incorporating this, we have the DNA polymerase fix the issue. It replicates the DNA, and then from there, we still end up with a little gap. And what we end up using to finish it off is a DNA ligase. So that's a base excision repair where we basically just get rid of the base. We then take the rest of it apart, like the sugar backbone, and then we replace it completely. So the difference between base excision and mismatch is that mismatch, we're going far upstream, whereas this one, we're kind of already like directly pinpointing exactly where the mismatch repair is and fixing it right there. Does anyone have questions about this one? Yeah.
Sorry, but it's the reason that we use DNA ligase for base excision and not mismatch because you're using a polymerase 1 and not 3. Like 3 doesn't need a ligase.
Mm-hmm. Yeah. In terms of ligase, that's a good question. Because theoretically, yeah, it should use the ligase here on this one. I can double check at the very end and see if it does use the ligase while these guys are presenting. I'll look it up. But I'm pretty sure it would use it, but I'll double check. Any other questions about this one? Okay. So moving on to the third one, we have nucleotide excision repair. So this one is specifically for DNA lesions. Remember those thymine dimers, the UV irradiation? We're probably going to use this type of repair rather than the other three to basically do a very crude cut around the DNA lesion itself. And I use crude because it's very far away from the DNA lesion. You wouldn't be directly on it. You'd be further away. And you cut it at both ends, hence why it's called exonuclease. It makes two cuts, and essentially it's going to cut on the farther end and then on the downstream end. And when it does that, you're going to use a helicase, and all the helicase is doing here is just disrupting the hydrogen bonds between the DNA itself that we want to get rid of and then the template strand that's normal. And then from there, DNA polymerase 1 fills in the gap, and then it has the ligase to DNA polymerase corrects the issue, and then from there, ligase is going to fill that last gap at the very end. Again, I will double check with the first mechanism because I'm pretty sure it should also have it. But does anyone have questions about this third one, the exonuclease? It's pretty straightforward. You just make 2 very big cuts. You use helicase to get rid of the bad mismatch DNA lesion from it, the DNA strand that has a DNA lesion, and then from there, you just fix it all up with DNA polymerase 1. Okay. And then for the last one is direct repair. So this pathway is very expensive. It uses a suicide enzyme where this enzyme has a cysteine residue on it. It forms a thioester linkage on it, and essentially it's going to, this is the most prevalent example is if we have an alkylated base, right? We have our guanine that has been methylated essentially. And what ends up happening is this methyltransferase, which is the enzyme, is going to grab onto the methyl group and basically turn this oxygen from an ether to a carbonyl group. And when it forms a carbonyl, we repaired the base, and then what ends up happening is this enzyme becomes permanently activated because, again, it has the methyl group. To get rid of this is going to be very difficult. But that's direct repair. Does anyone have questions about any of the 4? Okay. And then I put some practice problems here. The answers are in the notes. Some of these were from the exam, changing the wording up, but some of them were. So these are good to just look at in terms of the kind of depth they're going to test you on.
Alrighty, let's talk about transcription and RNA metabolism. As we move on to these lectures, just continue marking differences between eukaryotes and prokaryotes. We kept the same color scheme. Also, try not to confuse elements from replication with transcription, because I think it's very easy to be asked to just identify where different names and elements and enzymes are found. And there are certain similarities of steps in both of these processes, but they use different machinery. So definitely keep track as we go through. Okay, really quickly, just to introduce genes and RNA. I'm sure this is something that we've gone over multiple times, but there are nucleotides within DNA strands that code for genes. And what flanks them are usually regulatory sequences, which are in red. And then from that, obviously, we can replicate, as we just went over, and we can transcribe as well. Transcribe requires mRNA synthesis. As shown over here, it's a single-strand messenger, and it transcribes sequences found in those genes. I want to direct attention to this diagram because it is important in terms of types of structures that RNAs can have. RNA is not always single-stranded. So again, be careful when things are being described as absolutes. It's not always single-stranded. They're not always single-stranded. They're not always linear. There are ways that they can be secondary, tertiary, quaternary, and we'll talk about a few examples where that exists. So RNA structure, I'd say the most important ones are probably the secondary structure right here, the stems and the loops, because there are a couple of scenarios that we learned on where those are necessary, which we'll go into. I wouldn't worry too much about where things like single-stranded helix or hairpin double helix occur, but just recognize that they do occur in nature. Some viruses and things like that utilize them. But secondary structure, I think, is the one that you should be able to find a cellular reason to why we need them. Central dogma, something that we're all familiar with, really. DNA to RNA transcription, RNA to polypeptide translation. But don't confuse this with the fact that every single sequence of nucleotides goes to protein. Really, it's only 1% of the human genome is expressed as proteins, and those, remember, are flanked by a lot of these regulatory or non-coding sequences. There are 3 core rules of transcription. Number 1, a topic that we did talk about when we talked about replication, is that polymers are built continuously, and this is the word again, processive. We want to be able to keep doing this without our enzyme just falling off the strand, right? That was just not energetically favorable or favorable time-wise for the cell if an RNA polymerase is just falling off. Number 2, RNA sequences are complementary to the template strand, just like as we would expect. But RNA contains uracil to adenine bonds instead of T and A. And then lastly, RNA synthesized 5' to 3'. That's a really big rule of thumb. and that makes them anti-parallel. This is something that is pretty, I could see likely on the exam in terms of being able to note what's the template and what's coding. Rule of thumb, if it's a little trippy, the RNA transcript is exactly going to be the same thing as the coding strand. Like the only difference is the T and U. So just look for everything else that should match up with another strand that's coding. What doesn't match up at all is the template strand. Just to sort of denote what's coding and what's non-coding. And non-coding is synonymous with template in the case that they use that terminology. And the coding strand is coding not because it's what is being used as a template, but just because that is what your mRNA is, it's what it's expressing. It's the coding nucleotides. Alrighty, so in essential components of a gene, you should definitely be able to label where each of these terms exist on a strand of DNA. Starting with the promoter on the very left, reading from left to right, or really in your 5' to 3' direction. In the case that this was flipped, you should be able to place the promoter region in the appropriate strand, because either one of those strands could be used as a coding strand or a template strand. So depending on which one is given to you, the promoter is going to be closer, it's going to be on the side where your RNA polymerase is able to go in that 5' to 3' direction. So that's where your promoter needs to be placed. So don't always start left to right. In the case that we are left to right, just for sake of convenience, it's going to flank the transcription start region. We keep moving the 5' to 3' direction. We have our gene itself that's coding in blue. And then transcription ends at a location a little further away from what's actually coding. The start and stop aren't the exact nucleotides at the beginning and end of the coding region. There is a little bit of buffer. And that's important because if in the case RNA polymerase falls off a little too early, or it doesn't add the right thing, you'd rather it add a wrong thing in the place that's going to be chopped off anyways, right? Not in the gene where our important nucleotides are. Their functions are as said here. One thing I want to note is that be able to recognize what is a sequence and what is a protein or factor. Like a promoter is not a protein. There is no physical thing that's called the promoter that is going to bind here. The promoter is for your transcription factors and your RNA polymerase to be attracted to. On the other hand, something like a transcription factor is actually a protein that's able to navigate between multiple chromosomes and attach to its sequence, its promoter sequence. So there's a couple other factors and sequences later on in these processes that we'll learn about. Make sure you know what is a sequence and make sure you know what is a protein. RNA polymerase is obviously a protein. It's going to be catalyzing the synthesis of RNA. And the terminator, again, is just a sequence. There is a small exception in that in eukaryotic transcription, there are terminating factors that sort of help navigate that process. But for the sake of prokaryotes, it's just a termination sequence. OK, let's start with transcription, but we're going to start with prokaryotes first. Here's the Holy Grail diagram. Just to summarize the 4 steps, we assemble, going from closed to open. This is where I want to emphasize, especially the differences between going from closed to open in replication. It's not the same elements being used. So you can't confuse using the helicases and the gyrases that we saw in the replication slides with what's going on here. It's going to be a different type of opening and lessening of that torsional strain. Or not really torsional in this case, sort of just the affinity of the 2 strands, being able to loosen them a little bit to read. Phase 2 is initiation, then elongation is pretty standard, and then termination is where another difference occurs between prokaryotes and eukaryotes. There's a lot of different factors here. A couple I think you definitely should know. For example, the sigma factor and these elongation factors. Don't lose sleep over remembering NUS-A as an elongation factor, but know that they exist as these proteins that can associate and dissociate. The reason why I mention that is because this shows the idea that there's a lot of moving parts. Manipulating either one of those parts can affect the process. So you need to know at least where these factors are being placed along these 4 steps. Be able to know what part it affects. For example, if the elongation factor can never disassociate from RNA polymerase, Like, can you ever terminate? Probably not. And so that's the type of things you should ask yourself when thinking about a complex that is associating and disassociating. Yeah. Okay, number one, assembly in prokaryotic transcription. Two major things, or rather three. We have obviously our transcription factors, our polymerase, which is going to add those RNA nucleotides, and then the sigma factor. Transcription factor is that first main thing that signifies to the cell that, okay, this is the proper DNA that we want to read, and it's going to be attracted by that promoter region. The sigma factor is sort of like this assisting protein that is going to find specific DNA sequences within the promoter region itself. And these are called consensus sequence. So what that does is that it increases the affinity of RNA polymerase even more. Transcription factors already do that a little bit, right? They prime that DNA molecule to be attracted by RNA polymerase, but this is just another added layer of increased affinity. And so those two work together, and you could imagine that if a sigma factor wasn't present, you might not be able to get enough transcription required by the cell. You might get some RNA polymerase to lock in and read some DNA, but maybe not all the cells will have that mRNA that you care about. So you need both for the most effective transcription. You don't worry too much about what nucleotides are present here, but know that the consensus sequence is existing within the promoter. After that, we have initiation. And initiation also occurs at the promoter sequence. And that, like what this is describing is like going from a closed to an open state. It's just this like slight separation of strands that's very controlled. Like we don't want to separate them to the same extent as we did in DNA replication because that opens up a little too much DNA that we don't really need to be reading at the moment. We just want to open up the part that our transcription factor is indicating to us. Yeah. Here, I think this is where I emphasize that. Like, notice here that we don't really have our like gyrases or helicases here. It just seems like RNA polymerase and sigma factors help with this localized unwinding. Elongation, pretty standard. By complementing the template strand, we're able to replicate our coding strand. Again, remembering that there is no T nucleotide, only U. The rest of the three stay the same. Here, what's important is that your sigma factors disassociate, right? There's also like the idea of if your sigma factor can't disassociate, where are you stuck at? You're stuck at initiation. So you can't even get transcription in the first place. So that needs to dissociate. Number two, you need an elongation factor to maintain processivity, which is a characteristic that we talked about in RNA synthesis, we talked about in DNA synthesis. And so again, be able to describe where and which processes a word like that is important in. Just a closer look into active site. Like we talked about in DNA polymerase, it's not necessary to be able to draw this from memory, but just to be able to acknowledge that it's a similar trope, that you're bringing in this very large, bulky, negatively charged, probably unstable molecule to active site, right? So you need a lot of coordination there to keep that in place and make sure that it's added and not just lost to nature again. So that's why the active site here also has these aspartate residues, these metal ions, to make sure you're coordinating this phosphodiester bond in the right way. Now we have termination, and there are two main types. One is called rho-dependent, and one is called rho-independent. And I was a little confused when I first learned this for the first time, but rho is literally actually just the name of the helicase protein that is being used in rho-dependent termination. Rho-independent termination, this is the first time we now see this secondary structure of an RNA. So when the RNA is able to self sort of form into this secondary structure, it forms like a little physical blockage. It doesn't allow it to keep being read by the RNA polymerase. And so this hairpin loop is going to be formed when we hit that terminator sequence and then not allowed to be, and not able to go through the RNA polymerase once again, and therefore it will dissociate by itself. Contrast that to rho-dependent termination, which actually requires a helicase to do that whole dissociation for you. That helicase is going to bind in a region that's closer to its terminary sequence, and it's going to physically separate the mRNA from the DNA template for it. We don't have to worry about what the terminator sequence is or what nucleotides exist within it, but recognize that in one case you form a secondary structure, the other you stay linear, and your helicase does the disassociating for you. Let's go into a few differences. This is a diagram that represents the eukaryotic transcription method. And while there are a lot of similarities, and the 4 major phases that we talked about are the same, what the key is here is that there's actually just more factors that are being involved. And that's because eukaryotic transcription is a little more complicated. We have a lot more material. We have a lot more complexity in our genome. And so you want to be a little bit more regulated and controlled when we're working with transcription in eukaryotes. Because of this, you might have more transcription factors. What does that do? Transcription factors, remember, increases affinity. When we have more than one here, then we're really making sure that our RNA polymerase is on the right gene and on the right promoter. That's like factor number 1. In addition to that, there's also these termination factors that is different than what we had in prokaryotes. We had that termination sequence, right? This case, the terminator factors are yet another type of protein that's able to associate and dissociate when you want to release your finished mRNA from RNA polymerase. Last key point that is really important in terms of regulation is the C-terminal domain. C-terminal domain is sort of like this tail that extrudes out of RNA polymerase, and it's sort of like a sensor of where you are in those 4 steps. To continue on that, this diagram here shows this a little bit better in that on this tail, that is, you can imagine how, like, it's as long as you want it to be for the sake of our class, but it has a bunch of these residues that can be phosphorylated. Depending on the pattern of phosphorylation, the cell understands what phase of transcription you're at. So if these 2 residues, the very top, were phosphorylated at one given point, that means you're in initiation. In order to move on to the next stage, you need to change that pattern of phosphorylation on the CTD to indicate to your cell that, hey, let's recruit some elongation factors, and let's get rid of our sigma factor. And then later on, when we get to this terminating, or this phosphorylation pattern, let's get rid of our elongation factors, and let's recruit our terminating factors. So this pattern of phosphorylation is like a sensor. It's going to recruit what you want and get rid of what you don't need anymore. Zooming out a little bit, just want to remind ourselves that there are different types of RNA polymerases in eukaryotes, just like there are different types of DNA polymerases. The one that we're the most concerned about right now is RNA polymerase 2, which I know is annoying because DNA polymerase 3 is the one that we use in replication. It's not 3 again, it's 2 for transcription. RNA polymerase 1 deals with rRNA, which is relevant in the structure of ribosomes, and then RNA polymerase 3 is relevant for tRNA, which is important for creating proteins and peptide sequences. Any questions on CTD? Cool. Let's talk about a few examples of inhibiting transcription. Unfortunately, this is kind of like just thrown at us and just a memorization-based slide. But if there's anything I can emphasize here is probably the death cap mushroom. I think that's the one that if you have time to remember, maybe do that one. But broadly, also just understand where in the process that we just talked about can you probably get really potent inhibition. For example, there are things like rifampin where it blocks promoter clearance. That means we can't even get past the initiation state. And so that is one mechanism that's probably going to stop transcription in all cells. Over here, amantinin is very highly specific for the active site in RNA polymerase 2. In that case, sure, maybe it could have reached the promoter site and start elongation, but it has no space left in the active site to bring nucleotides through. So that's another mechanism of inhibiting transcription. So at least be able to explain what the mechanisms are and sort of what the general scientific idea is behind these inhibitors. After this, just like we talked about in telomerase, there are these enzymes called reverse transcriptases. And so another example, in addition to telomerase, is especially in like viruses, they have figured out ways to effectively target host cells and infect host cells by bringing in RNA molecules along with enzymes called reverse transcriptases and using those RNA templates to build viral DNA. And then that viral DNA will be, read. Like it sort of tricks your cell's machinery to read the viral DNA and then cause cell death, cell lysis, and whatnot. HIV is an example of a virus that utilizes reverse transcriptase to sort of bypass the cell. Because DNA is often problematic. We don't want just random DNA getting through. It seems like RNA can get through that first checkpoint, and then the cell is sort of tricked into reading it, and that causes a lot of infection. This is an example of an analog called AZT that can inhibit reverse transcriptase. Notice here that it looks very similar to nucleotides that could be involved in creating RNA, creating DNA. But structurally, look at this little trio of nitrogens right here. That doesn't usually exist in our nucleotides. And so while it is very similar, if reverse transcriptase was to pick that up, it can't keep adding on after that point. So it sort of inhibits the activity of reverse transcriptase because of the structural difference. Let's talk about post-transcription. Change the color to pink because now we are in exclusively eukaryotes. Prokaryotes don't have post-transcriptional processing that at least we're concerned about in this class right now. There are three major things. Number one, the 5' end is capped. The 3' end has a poly A tail. And the middle portion, there's some splicing going on that makes sure that we only keep the part of the gene that is coding for a protein, and not really the intron in the middle that's going to go away and be degraded by the cell. A few key points here. Number one, obviously, does it happen in prokaryotes? The answer is no. Number two, they happen co-transcriptionally. Think about why that's important, right? We want to save time in all these processes, so while your mRNA is being made, might as well start adding that cap and working on some splicing at the same time. Yeah, is there a question? No? Okay. Cool, let's talk about these a little closer. Number one is the 5' end. I would try to have these enzyme names down, at least the guanylyl-transferase, because the key here is that when we're adding that G, this sort of modified G as a cap, so at least recognize the enzyme name that's at play here. They're capped for two major reasons. One, to protect from ribonucleases, because where's a lot of our, not all of our DNA, but our RNA, but where's a lot of the mRNA going? It's going from the nucleus outside to various organelles and cytoplasms. Cytoplasm, however, has a lot of these nucleases that's going to want to degrade mRNA, just because it doesn't know any better. The cell doesn't actually know it's mRNA unless you add a signal that saves it from those endonucleases. And then number two, it's also to enable binding to other proteins and ribosomes. We'll talk about this in a future slide, but signal sequences are sequences that sort of attract various items, elements to different cellular locations, and the 5' end is actually involved with a lot of that. Okay, let's reach the right location, and let's get bound to the right thing. So it's a multi-pronged reason. And again, just to emphasize, it happens at the same time as you're transcribing that full length of mRNA. Splicing, so now we're moving sort of towards the middle of the mRNA, or the immature RNA at this point. This is the mechanism of splicing, and there's different ways. You can either be self-spliced, or in the next slide we'll see a protein helping you out. I think it's like I wouldn't spend too much time like trying to figure out the arrow pushing behind splicing. But what I do want to emphasize is that you can't just splice anywhere. There are specific splice signals that are necessary at one end of that exon and the other end to indicate where that splicing needs to occur. And so it's also a very regulated process. And if you want to go just a step deeper, these signals are usually indicated by a G and an A. And it's the nucleophilic attack of this A that's going to sort of cut the intron outside of itself, basically. And so just recognize that you need those signals there in order for splicing to occur. Whether you're doing it self-splice like this, or whether you're using an element or a protein that's going to do that cutting action for you. And again, this is going to conjoin your exons and let your intron in the form of a lariat loop to be degraded. And there's no ligase needed here either. Even though we're trying to conjoin two segments by cutting one out, we don't need a ligase, as we did for various repair mechanisms in the last lecture. Similar in the enzyme-directed version as well. Don't stress too much about all these different components. The thing here is that because you require so many protein components to assemble on either side of the intron, the issue here is that this is ATP dependent as well, because you need to coordinate that protein activity somehow, and you can't just do it the RNA can't just do it by itself, but it accomplishes the same exact thing. You form a lariat from your intron, and you conjoin your two exons without a ligase. But don't worry about the different U factors. Now moving on to the very end of the mRNA, the 3' tail. I would know the enzyme names for sure for these. There are only two, and they are quite important. The first one is endonuclease, because the key here is that there's a little bit of buffer at the end of the primary transcript that we don't really need. And again, this is a very smart decision on the cell. It purposely adds a few more things to be coded because it is a lot more riskier if you ended right here. This right here is your consensus sequence that indicates that you want to add a 3' end. If you ended your transcription here, there is no guarantee that some of these nucleotides won't fall off. You'd rather have nucleotides fall off when we don't really care about them. So a little bit of this cushion is necessary, because then your endonuclease is going to be able to read that, okay, this is just fluff material. It's not actually my consensus sequence. I'm going to cut it just so that the consensus sequence is left. Once the consensus sequence is left, polyadenylate polymerase is going to go in and add that poly A tail. Not really sure how many poly A's or how many A's get added. Depends on the species. But similar reason as the two that we learned about the 5' cap. One, to protect the mRNA when it gets transported somewhere else. And then also there's some signaling property to that as well. It can sort of navigate the mRNA to the right place. Okay, just a summary slide of mRNA changes, post-transcriptional changes. Again, this happens in eukaryotes only, and I think it is safe to use the nomenclature of mature and immature. Immature suggesting that you haven't done these changes yet, and mature that you've done all three. And that mature mRNA is what's going to code for a protein product. A little question that I just wanted to ask, just to think about. If you need to read an mRNA that's given to you in a sequence of nucleotides, there's a really quick shortcut now that we've learned to figure out what the polarity of the sequence is. And that's now our 5' and 3' cap. If you saw a bunch of A's at the end, then you know that must be the 3' polarity. And you saw a bunch of you saw a G at the very, very end that's methylated, then you know that's the 5' end. So from there, if you go back to that slide that had those three strands, the coding, non-coding, and the mRNA, now with those extra bits of information on the mRNA, we can work our way backwards and figure out what was the polarities of our coding and non-coding strand. So look for clues like these when given a bunch of nucleotides, because I know it can just be kind of just like a word dump when seeing a bunch of combinations of 4 letters, but look for these things that we know indicate that the process is done. And the diagram on the right is just appreciating the fact that you can alternately splice. Prokaryotes can't do that, but eukaryotes have the ability to make one primary immature sequence into various mRNAs. And that is seen as therapy for a disease called spinal muscular atrophy. Key here is that there is just a gene called SMN1 where its protein product is what you need for proper spine muscle function. In SMA patients, however, it seems like the SM1 gene has a mutation that doesn't allow it to create that proper protein. So what scientists have figured out is that they can sort of exploit the SMN2 gene and then induce splicing such that all of these gene components that were in SMN1 also exists in SMN2, and therefore we still get proper protein product. If you're inducing, you can induce alternative splicing to get what you want, is the key here. Now really quickly to go over ribosomal RNA called rRNA. Again, this is from your RNA polymerase 1. And don't worry too much about the steps to create it, but recognize that it needs to be cut up a little bit because our ribosomes are made of differently sized subunits. And so you have multiple types of rRNAs making up a ribosome protein. And the unit it's called S. I think this is relevant because you could be asked to sort of look at a graph or a chart or a gel that has separated these proteins and point out, based on what we know about ribosomal structure, what could possibly be the band or the segment that makes up the subunit of the ribosome that we care about. The next lecture will have a couple of those subunits that we commonly see in a ribosome. We also have tRNA, formed by your RNA polymerase 3, and we'll talk about that a lot more in this next lecture. You can regulate gene expression with mRNA as well. It sort of tells us a little bit about whether we want to be in a synthesis mode or a degradation mode. When synthesis equals degradation, mRNA concentration remains the same. And the same idea of if you have like a lot of synthesis, or when you have an mRNA, you might have a lot of synthesis or being pushed towards a lot of synthesis. And then when you've used up all that mRNA to form protein content, it's time to degrade all of it. It's sort of just the idea, and the other way around as well. If you don't have any mRNA, you want to be pushed towards a state of synthesis and start making those proteins up. MicroRNAs are also really important as an inhibitory mechanism. We haven't learned too much about what inhibits mRNA production, besides the fact of like, okay, if you already synthesize enough, why would I want to keep transcribing? But this is a more direct mechanism. This is a sort of, there are two ways that microRNAs work. One is that it can directly degrade the mRNA that was formed and regulate its translation that way. Or what it can do is it can sort of indirectly inhibit the ribosome from reading that mRNA. Basically, all you need to know is that it's a 22 nucleotide piece of RNA. It is almost perfectly complementary to the mRNA that it's going to target and inhibit. And don't worry too much about this dicer and risk, but I would know the two general mechanisms by which you can inhibit gene expression processes. And one of those is by just literally degrading the mRNA, and the other one is just not letting it go through ribosomes. So what this begs the questions of is, what would happen if you didn't have a mechanism like microRNA? You didn't have a way to directly regulate transcription and translation. That is also not good for the cell. You don't want too much product always. Don't want too little. And also think about it from the perspective of the microRNA. What if I create too much microRNA at a given point? Then I'm probably not going to get any protein product that I care about. It's a very, very intricate scale. Ribonucleases are the final way that mRNAs can be degraded. Obviously, at one point, we don't really need the mRNAs because we reach the protein content that we care about. Prokaryotes do it a little differently, involving a lot more RNases, whereas in eukaryotes, you reach this structure called an exosome, which has various components that I would just broadly know. You start with the helicase, you go to an ATPase, which requires energy. The energy is going to be channeled toward the exosome core, which is attached to a nuclease that does the actual cutting. I wouldn't worry too much about the steps, just recognizing that there are different mechanisms at play here. Okay, before we go to protein metabolism, anyone have any questions on transcription? Okay, let's talk translation. Here is the genetic code. This is a chart that you will receive on the exam, so you don't have to worry about it. Besides a few exceptions, I think you should absolutely know without reading the chart what's your start codon and what are your few stop codons. That is unlikely to like the start codon and the stop codons. It's not necessary that they indicate that on the codon chart. So just be able to point them out yourselves as well. During translation, the key here is that 3 mRNA nucleotides are read at one time, and that produces one amino acid, or that brings in one amino acid to your growing chain. Here, I just want to mention that you should understand the concept of degeneracy or codon redundancy. Be able to explain that one amino acid can be coded by multiple codons, and that's sort of just an efficiency thing here that just allows things to be just a little bit quicker when you translate. Just like transcription, there are a few important locations on the single strand of mature mRNA that we care about. The core rules still exist. We go from the 5' to 3' direction. Translation will start at a start codon, AUG, methionine. Honestly, if I was to really say, I don't default to methionine because we see that in prokaryotes, it's not just methionine, it's this F-methionine that is a slight nuance between the two, but obviously, overall, it's okay to think of it as methionine. We go through in the 5' to 3' direction, and then we stop translation at the stop codon, and there's a few of those. Think about here, what happens if you don't have a start codon? What happens if you don't have a stop codon? What happens if you mutate in between these two areas, the stop codon? They're all not good things. Before the specifics, let's go through just the overall pockets of a ribosome. Translation moves in the 5' to 3' direction, and so I would encourage you to read the diagram backwards. We really can't read it as EPA, but rather it's APE. You enter at the A site, or your tRNA binds there. It elongates your existing strand in the P site, and then it's able to exit at the E site. This is what I was talking about with the two subunits of ribosomes. There's a small one and there's a large one. Small one often binds to the mRNA, and the large one, because tRNA, as you saw, had this multiple loop structure, is a little bit bigger, so that tRNA could properly lock in and bind, or not bind, but rather be able to read just the codon that's right in front of them. You don't really want it to be loose. You don't really want it to move around too much in that area, because it should only read the three nucleotides at a time. Some broader structure, ribosomes are highly conserved around organisms. They're large and small. Prokaryotic have the 50S and the 30S respectively for large and small, while eukaryotes have slightly bigger ones, 60 and 40. I think you should know absolutely that there are two subunits, one large and one small, but don't lose sleep over not knowing what the numbers are. Okay, let's talk about tRNA, how it looks like. A lot of different labels here on this diagram, but the most important one is at the very bottom in green, and that is the anticodon. Because just like our RNA polymerase reads a template to create RNA strands, this is sort of the backwards now. Like, we're reading our RNA strand with an anticodon. Because the three nucleotides in RNA are the codon, but tRNAs have its complementary nucleotides also going in the 5' to 3' direction. And so that is how the tRNA is going to be able to read the nucleotide or the codon that it cares about. Other arms are like the two on the left and right. I wouldn't emphasize too much. I think they're more as structural units. Make sure that the tRNA is stable, able to fit into that large subunit. And the one on the top, also in green, is where the amino acids are going to be added on and elongated. Key here is that I think this diagram is a little misleading in that it sort of suggests that one tRNA is being used the entire time to keep adding amino acids, but really it's a different tRNA brings in an amino acid each time you go through elongation. So it's not just one tRNA doing the whole job. Let's talk about wobble hypothesis really quickly. I think this is super important to be able to explain. Try to practice explaining this in like a sentence or two, because conceptually it is very important. And it's not just a bad thing. Like, wobble hypothesis sounds a little like not good, or it doesn't put the tRNA in good light. But the wobble hypothesis is important because it helps with speed. The idea is that in the wobble hypothesis, the very first position of the anticodon, first is synonymous to 5' position of the anticodon. It has this wobble characteristic in that sometimes it might be edited to this other base that is not generally used in a lot of our nucleic acids called inosine. You might have remembered inosine from one of our synthesis pathways, but inosine has this little it's not as specific as the nucleotides that we're generally used to. As you can see here, it can bind to various things, and what that provides is that redundancy to our codon chart, basically. As we can see here, because inosine can bind to various things, we only need 32 unique tRNAs for all the codons that we saw on the chart. You don't need 61 different tRNAs for each of the codons that they might encounter. And therefore, this helps with speed, and it also helps from a structural standpoint as well. Imagine that every tRNA absolutely needed its 3 complementary nucleotides to its codon. What you build there is risk of not being able to disassociate your tRNA. So what the wobble does is that it helps ensure that we can disassociate our tRNA when that amino acid has been brought and added on to the existing chain. Just a multi-pronged reason, one for speed, one for making sure that we can keep the processive nature of translation. Okay, so we have our mRNA. Let's decode it now. This is the reading frame. The reading frame is important because it starts at the start codon, goes away to the stop codon, and the reading frame is not always set in stone, can be affected by various mutations. AUG is our conventional start codon, goes from methionine, and then the stop codon, the 3 major ones are here, UAA, UAG, and UGA. Again, emphasizing, just like I did in RNA synthesis, is that it's not necessary for them to give you an mRNA sequence that's in 5' to 3' direction. Don't panic if you don't see AUG in the very beginning. It's fully possible that they'll give you an mRNA that's a lot longer than what it needs to be, but the AUG might exist somewhere in the middle or a couple codons after. Just a way to sort of make sure you know what you're looking for, and you're looking for that AUG. In addition, it might exist on the right side of that sequence, and I know we're conventionally used to reading left to right, but if the polarity is given 5 to 3, you need to look for that AUG on the right side of your page, not the left. So mutations that can affect the reading frame, or even not, or the ones that affect them are going to be on the bottom, insertion and deletion. Again, we read by 3s, and therefore inserting one or deleting more, or inserting one or more, or deleting one or more will affect the reading frame. Interestingly, though, for example, there could be times where you add 3 at a time or delete 3 at a time. Maybe then you won't affect all the preceding or sequential codons. All you're going to lose there is just one of those things, right? In other cases, if you add just 1 or 2 nucleotides or remove 1 or 2, then yes, you disrupt the by 3 nature of the mRNA. Things that don't affect the reading frame broadly are things like silent mutations, missense mutations, nonsense mutations. Just be able to look for those in an mRNA sequence. Be able to compare the original strand and the mutated strand and point out that, hey, a silent mutation is that you're not changing the amino acid sequence, even though you're changing the codon sequence. Missense being that you had this one mistake that just forms another codon and therefore another amino acid. And then nonsense being that you made a stop codon a little too early. And therefore you won't get the protein that you want, or you might get a protein that just can't fold properly. Okay, now let's physically build our amino acid sequence, and now the EPA or the APE region is important. So here's our primary diagram. Once again, that keep track of eukaryotic and prokaryotic differences. This diagram is in the setting of prokaryotes. And the number one thing that you can use to figure that out is a Shine-Dalgarno sequence. It's only used in prokaryotes. We didn't talk about this in the class about what eukaryotes use, but they have a little bit of a different mechanism to figure out where and when you need to start translating. And so the steps overall is that you need to activate your amino acid, starting over here. You have to initiate by docking your tRNA at the ribosome. Then you got to elongate by having these processive cycles of tRNA bringing your amino acid, adding it to the pre-existing chain. And then while this is all happening, the protein can start folding into its secondary structures as well. And then once it's fully disassociated, once it reaches that stop codon, then it can have all those proper 3D folding that needs to occur. Let's start looking closely at tRNA activation. The primary thing here is that you need to be activated by having an amino acid already on the tRNA before you bring it to the ribosome. It's not really like it's a very stepwise procedure, and you can't just bring a tRNA and dock it on the ribosome and then add your amino acid. That requires a little bit of energy as well. Class 1 of tRNA activation, you have the 2' OH attacking the amino acid and then transferring it to 3' OH. Class 2, the 3' OH attacks the amino acid directly. And this is the 3' and 2' OH respectively on the tRNA, specifically the end that's going to carry the amino acid, which again is at the top of the molecule. What's at the bottom is that sort of like that loop at the very bottom that's going to have that anticodon region. For initiation, now we see the existence of a few more factors and sequences. Again, to separate, Shine-Dalgarno is a sequence, and initiation factors are proteins. What's going to happen here is that we only have our small subunit of the ribosome right now. This is still in prokaryotic, so it would be the 30S subunit. That plus the initiation factors are going to all bind to the mRNA. The Shine-Dalgarno sequence of the mRNA is going to bind to a specific portion on the small subunit. I wouldn't necessarily also focus on the 16S portion, but just recognizing that it's the small subunit that we're binding to first. Importantly here as well is that this is not just a normal methionine that we're used to in prokaryotics. It's a F-methionine, and that's going to bind, or the F-methionine that's bound to the tRNA is going to bind to the start codon. Not the F-methionine itself. Again, it's the anticodon region right here. Keep in mind that if this is going 5' to 3' left and right, the mRNA, your anticodon is 5 to 3 in the backwards direction. And then once that has docked on properly, you can add in your larger ribosome subunit. Once that large subunit is bound, then you can dissociate your initiation factors. And then that subunit will stay bound to the small one and then continue or allow the elongation process to occur. Wouldn't worry too much about these individual initiation factors like 1, 2, and 3, or the steps that are required to get this. But just recognize that it's small subunit first, along with your mRNA and your initiation factors, and then you add the large subunit when you're ready to start elongation. The key difference before we get to elongation in terms of eukaryotic translation is that we don't really have a Shine-Dalgarno sequence. Looking at the diagram on the left, it's actually the 5' cap and the poly A tail that navigate the mRNA in the right place along the ribosome to start translation, instead of a specific sequence. And then also on the right, we're not adding an F-methionine amino acid to the tRNA. We're adding it to the regular methionine that we're used to seeing. And so, again, this is a specific example of why the 5' cap is important in addition to protection. Okay, let's go back to, I think these diagrams are in the prokaryotic context, but really now we're getting a little bit more generalized. What's happening here is that a new tRNA is going to bring the second amino acid in sequence into the A site of the ribosome, the larger ribosome subunit. And this requires an elongation factor called EF-2. I would know the EF2 part because the elongation factors differ when we get to another step in the process. Here what happens is that in the A site is where the two amino acids that we want in sequence are close enough in proximity in order to form a new peptide bond. It's not super relevant to know the error pushing behind that mechanism, but recognize that this is as important as it is it's also a very localized and proximity-based process as well. Like these EP and A sites couldn't be any farther than they are right now, or else this process or this mechanism of adding a peptide bond wouldn't be possible. This peptide bond is added in the P site, and once it is added, the tRNA that was already there, holding that first amino acid, will leave while the one that brought in the subsequent one will take its spot in the P site. So while you're still elongating, there is going to, a tRNA will remain in that spot. There's only flux into the A site and out of the E site, sort of pushing the one that was already there out of its place. Cool. And then lastly, I guess, just for, I don't know, an extra fact is that what allows the tRNA to dissociate finally from the E site is GTP hydrolysis, because energy hydrolysis causes some conformational change that gives it some room to disassociate. So yet, this is now another place that we see some possible regulation or targeting. We learned about this as sort of the GPCR protein or signaling pathway, right? That when we take away that pathway's ability to hydrolyze energy, what happens? We can't disassociate or we can't regulate the pathway properly. Similarly here, what if we didn't have the ability to hydrolyze GTP? Then you would never be able to leave the exit site and you can never keep adding your amino acids. So this is another place that you can potentially think about inhibiting translation. Termination requires slightly different factors. When we're happy with our polypeptide sequence and we've reached our stop codon now, we're going to have a different elongation factor arrive. In this case, first off, we have a release factor, which is able to bind to the UAG. I'm honestly unclear if there's a way for the release factor to read this stop codon. We just didn't learn too much about the structure, but something is reading that stop codon first. When that release factor binds to the stop codon, you are able to first pull apart the polypeptide sequence from that already existing tRNA. And then lastly, you bring in a component called EFG, which is not the same as EF2, and that finally includes the last step of GTP hydrolysis, which disassociates the large subunit from the small subunit. And so that is, again, another example of when hydrolysis is used for conformational change. In regular elongation, the only thing that GTP hydrolysis is allowing your already used up tRNA to leave and go do other things. This case, this final step of hydrolysis allows the ribosome to disassociate fully. Let's talk about protein folding. Just to remind ourselves that this happens spontaneously and co-translationally. So as this blue polypeptide sequence is being formed at the very top at the P site, it is able to undergo its secondary interactions and start forming those tertiary bonds. Once it's fully dissociated, right, at termination, I think it gives it a little bit more room, obviously, to form into its 3D structure, quaternary, even if it needs to. But there are a few post-translational factors or modifications that could occur. There are a couple examples. We didn't learn about them as in depth as we learned as the post-transcriptional factors, but be comfortable listing a couple. And I think if it's easier for you to remember them from their function. Like, for example, we don't want proteins to just stay in the spot that they're synthesized in, right? We want them to go to multiple different places. So one thing you can add to them are factors that allow them to localize to specific organelles. Number one, you can add lipid chains on them, because maybe lipids allow them to be stuck to the center part of a plasma membrane, right? But also, we also learned that if any of you guys have taken physio or other classes in the bio department, not all proteins are active right away. They require cleavage into a smaller form in order to have actual active biological function. So those are three possibilities. Post-translational factors. We haven't learned mechanisms behind any of them, but I would know their physiological function at least. In terms of inhibiting, again, sort of just a memorizing slide, unfortunately. I would try to remember the names if you have time. But again, know the mechanisms first. What you can do is that you can block either site of the ribosome. Imagine what would happen if you block the A site. You can't even bring a new tRNA. Imagine if you block the P site. You can't even add a new one. If you block the E site, you have no clearance. So all those are not good. If you block tRNA binding, you can't even add a large subunit on top of it. Because remember what happens first. The small subunit needs to bind to the tRNA. Then only you can add the large subunit. So that stops you from doing anything in the very beginning. And then end all be all, what if you just impair the ribosome structure? You can't do any of the two that I just mentioned. So understand the mechanisms, be able to create a plan, not a plan, that sounds kind of like evil, but be able to describe experimentally what could you do to inhibit a ribosomal activity. Lastly, I talked about this a little bit, but there are these things called signal sequences that, in addition to post-translational modifications, can indicate to proteins where they need to go in the cell. Nuclear localization signals direct proteins to the nucleus. They're aided by these things called importans, because they're being imported into the nucleus. That is sort of in the case of free ribosomes that exist in the cytosol. But there are also bound ribosomes that are found in the rough endoplasmic reticulum. And we've learned about the endoplasmic reticulum in previous classes, and we know that it's like the main exporting system of the cell. And so those consist of a lot of these signal sequences or signaling factors that allow our proteins to be transported to the right location. And lastly, while these are movements that are sort of like, okay, they're going to go on to do other functions, there's also movement involved in degrading proteins, and that's what lysosomes are good for, because lysosomes have very acidic environments. Very unlikely that a protein can handle that. And so when you need to degrade, you're probably going to shuttle them into a lysosome. When you want them to get somewhere that's important, you're probably going to put them in a secretory or vesicle. Just another diagram to show you sort of how the process works when you have a bound ribosome. This ribosome is connected to the ER via a receptor. And what's really convenient is that it sort of works in this loop formation such that while the mRNA goes in like this very cyclic manner, your amino acids are just added within the rough ER anyways. Lastly, let's talk about protein degradation. There are two major methods. Prokaryotes do this a little different than eukaryotes. This is a very oversimplification of prokaryotes, but I think that's all you need to know. One, I guess the one similarity between the two is that they're going to require a tag. And that's the idea of that you don't want to degrade anything at any given time. You got to tag both. We don't really know what the tag is in prokaryotics for this class, but in eukaryotes, we know that the tag is ubiquitin. Very important because it degrades proteins for degradation, brings them over to a proteasome, which, similar to the exosome, is going to degrade a protein. What I especially want to emphasize about ubiquitin, and its very long mechanism, don't have to worry about all these factors, the E1, E2. If you have time, please go over them. But what I want to emphasize the most is this little lysine residue that the ubiquitin finds to tag it. And why is that important? Because lysine, we know, is a charged molecule, and therefore ubiquitin is going to be looking with sort of these charged portions of the protein in order to tag to. It's not going to tag to any residue on the protein. It'd rather look for something that it knows it can find easily, and it does so by charge. Any last-minute questions on these lectures? Solid.
I'm going to refresh these just to make sure it's safe. Yeah. There we go. Cool. Okay, we got 2 lectures left. I'm going to start with lecture 22. That's regulation of gene expression. So why do we need to regulate gene expression? Professor Holmes had a slide up in his lecture that was like making a small protein take something like 1,200 ATP. Making proteins is expensive. It takes a lot of energy. It takes a lot of resources. So we want to be really sure about it when we are doing it. There's a bunch of different broad types of transcriptional control. Genes that are expressed only in response to stimuli are what we call induced. And genes that are inhibited until we need them are what we call repressed. When I took the class, I had a bit of trouble sort of conceptualizing that. So I think of induced genes as sort of off by default, and repressed genes as sort of on by default. So the example that Professor Holmes gave of induced genes are heat shock response proteins. So if a cell enters a situation, that is extremely hot, you're in conditions where you need to produce those proteins, so those genes are going to be induced in response to that situation. The example he gave for repressed genes was DNA damage repair, which we will cover in a lot more detail later. The last kind is genes that are expressed all the time. These are called constitutive or housekeeping genes. And then more broadly, there's two kinds of transcription factors that we'll talk about. So general transcription factors are transcription factors that bind to the promoter, and they help recruit our polymerase by increasing its affinity for the promoter. On the other hand, we have specific transcription factors, which is what we're going to spend a lot more time on today. Specific transcription factors are binding to regulatory sequences, either activator binding sites or operators, to control the transcription process. It's a text-heavy slide, but we'll walk through it. So there's two main kinds of regulation that we're going to talk about. Positive regulation is when we have activator proteins that are going to bind to the activator binding site. These are going to promote or increase transcription. The other kind is negative regulation. This is when we have repressor proteins acting, and these are going to bind to the operator and inhibit transcription. So you can see in these diagrams, there's sort of, for each of these, there's two different ways to do this. For positive regulation, you can either have a situation where the activator is on by default, and then a molecular signal is going to cause the activator to leave, or a situation where gene expression is off by default, and a molecular signal is going to cause a conformational change in the activator, which is going to cause it to bind to this site, this activator binding site, and increase transcription, promote transcription. The same goes for negative regulation. So you can have a situation where your repressor is on by default. Here, I'll circle it with this. On by default, which means that gene expression is off by default, and then a molecular signal causes a change which removes the repressor, which in turn turns on transcription. And the same thing where you can have the repressor off by default, and a molecular signal causes a conformational change which makes the repressor bind and turns gene expression off. That's a mouthful, but in general, know that there are different ways that positive and negative regulation can happen inside the cell. Another note is that in prokaryotes, our control elements, these operator sequences and activator binding sites, are going to be proximal to the transcribed regions. That just means they're pretty close to the regions we're transcribing. On the other hand, in eukaryotes, they can be proximal and they can also be distal. We'll talk a little bit more later about what this looks like and how we deal with that. We sort of wrap the DNA around in 3D space. But yeah, important distinction. Okay, operons. So operons are DNA sequences that are going to code for multiple proteins. These proteins are generally used in a common pathway. A simple way to think about this is that you have one DNA molecule that is going to code for one mRNA molecule that codes in turn for multiple proteins. These genes share a regulatory sequence, and they have the same promoter. So it's easy to control the expression of all of these genes from one place and in one way. Each gene, though, is going to have its own start and stop codon because we need those for translation. If we don't have those, you're going to build all the proteins together into one, which you don't want. Operons are what we call polycistronic. That just means that one strand of DNA is going to create multiple proteins. One DNA strand, one mRNA, multiple proteins. One of the most studied operons, and the one we are going to talk about in this class, along with the tryptophan operon, is the LAC operon. So the LAC operon is going to control production of proteins that we use to convert lactose into forms of energy that the body is more familiar with using. So if you remember, lactose is a disaccharide made up of monosaccharides galactose and glucose, which we both have. We have pathways for both to get energy out of those. So we want to break those down. Important genes that are encoded in the LAC operon are LAC-Y. LAC-Y is going to code for lactose permease. That's that sort of channel up there that's going to allow lactose to enter the cell. The other one, LAC-Z, is going to code for beta-galactosidase, which is what breaks lactose down into galactose and glucose. It's important to note that that step also allows for the isomerization of lactose into allolactose, which we will mention later on as well. The last gene that we're thinking about is LAC-I. LAC-I is going to code the LAC repressor. The LAC repressor has its own promoter, just a little note. Some other regions in this operon that we care about are the activator binding site, which we're going to also talk about later. That's this little blue region here in front of the promoter. And then we also have three operator sequences. That's O3, O1, and O2, which is where the LAC repressor is going to bind. Yeah?
Do we need LAC-Z to get allolactose?
That's a good question. No, I phrased that badly. You don't need LAC-Z to get allolactose. That's a good point. Yeah?
Do we care at all about LAC-A?
Not for this class, no.
Okay.
Okay, negative regulation. So we're going to talk about both negative and positive regulation of the lac operon. We'll start here. So the lac repressor is what negatively regulates the lac operon. Is going to do so by binding to two of these sites. So we have, it has a high affinity for O1, the O1 site, so it's always going to bind to that and either O3 or O2. If you look at this diagram right here, you can see that it's bound to O1 and O3. And when it does so, it's sort of twisting up our gene. It's going to twist up the promoter in this case, which means that no transcriptional machinery can bind there, and you can't transcribe that gene. The other option is that we bind to O1 and O2, which is going to it's going to cause LAC-Z to twist up. Can't transcribe that region. Similar situation. The lac repressor can also be repressed, which is confusing, but the thing that's doing the repressing of the repressor is allolactose. So allolactose is that isomer that we mentioned in the last slide. Allolactose is going to bind to the repressor and cause a conformational change in the repressor, which means the repressor will then have a decreased affinity for binding to DNA. It's not going to be able to bind to those operator sequences. Repression's not going to happen, and we can go on and express the gene as normal. We can transcribe it as normal. This is just a brief summary of what we just talked about. You'll see these in a couple of these slides in case it's helpful for studying. So we talked about negative regulation. We'll also talk about positive regulation of the lac operon. So as glucose levels are decreasing, we're going to form cyclic AMP or CAMP. That's the second messenger that we've talked about before in this class. CAMP is going to activate CAMP receptor protein, or CRP. CRP is an activator protein of the lac operon. So when you have high levels of CAMP, you're going to activate CRP, which in turn is going to bind to the activator binding site of the operon and going to promote transcription. Again, these are the overall steps. It's important to note that this is going to allow us to have sort of multiple different states, multiple levels of regulation. So you're going to have an off-on state, which is what we mentioned in this negative regulation area. And you're going to also have this sort of low expression and high expression state. The low expression state is when the gene is on, when transcription is on, but you don't have that activator bound, CRP. And the high expression state is when the gene is on, but you do have CRP bound. This is a sort of big summary slide. I think that it's very, very likely that you'll see questions on the test that are like, hey, you're in a state with high lactose and low glucose. How do you deal with that? What is made? What happens? I'll walk through this really quickly, but could be useful for studying as well. So if you have lactose and you also have high levels of glucose, you don't really have a great need for breaking down that lactose because glucose is the preferred energy source of the cell. Lactose absent, high glucose. We'll start with that. If you have no lactose and you have high glucose, you have literally zero need for this pathway. If you don't have any lactose, you don't want to make machinery to break down that lactose because that would just be a waste. So we are going to turn off gene expression. The lac repressor is going to bind. You're not going to be able to transcribe that gene. If you have lactose absent and you have low glucose, it's going to be the exact same situation. So repression is acting as an on-off switch. If you have lactose absent, no matter what, you are not going to transcribe that gene. On the other hand, when lactose is present, we're going to talk about the two right ones. When lactose is present and when you have high glucose, the presence of lactose is going to cause the formation of allolactose, which in turn is going to turn off the repressor. You're going to do gene expression. High glucose, though, means that you don't need to do that much gene expression because you have a lot of energy. You don't need to break down a lot of lactose. High glucose is going to mean you have low CAMP. You're not going to bind your activator. You're going to have low levels of expression. When lactose is present and you have low glucose, you're going to have a situation, again, where gene expression is on. But this time, because of low glucose, you're going to have a lot of your second messenger CAMP. You're going to activate a lot of CRP, which is going to bind to the CRP binding site, which is going to activate transcription even more. So we have these four states. We have off, on but low, and on but high. Any questions about that or the lac operon in general? Cool. Okay, the other operon that we're covering in this class is the tryptophan operon. So the tryptophan operon is going to contain genes that encode for proteins that we use to make tryptophan. This operon is controlled pretty simply by negative feedback. There's two things that we're going to talk about, two ways to regulate this. The first one, pretty straightforward, negative feedback. You have this trip repressor, which is going to bind to the operator sequence if trip is present, which in turn, it's feedback regulation. If you have high levels of tryptophan, you don't need to make more, so that's what we're doing there. The other way that we're going to talk about is a bit more complex. Important to note from this diagram here that we are coding for this leader peptide. The leader peptide contains an attenuator, and we're going to use that for modulating this pathway. Okay, so that leading peptide that we talked about, it codes for 2 tryptophans back to back. It has 2 tryptophan codons back to back. This is going to act as a sort of biological sensor of tryptophan. So if you have high levels of tryptophan in the cell, you're going to be able to make a lot of that charged tRNA with tryptophan, which means translation is going to occur quickly. If you have a lot of tRNA tryptophan, there's going to be no pausing when you're transcribing this region. You're just going to zip right through. And when you zip right through that region, that gives a lot of time for this 3rd and 4th sequences of your leader sequence to hang out next to each other. They're going to form a hairpin structure, and the hairpin is going to terminate transcription. We talked about that a little bit in the previous lectures. The other situation is when you have low tryptophan in the cell. That means that you're going to have less tRNA charged with tryptophan, which means that translation of that trip region is going to be slower. The ribosome is going to get here, and it's going to sort of pause for a second while it waits for tryptophan charged tRNA. And that's going to give time for 2 and 3 to form this hairpin structure. 2 and 3, that hairpin is far enough away from RNA polymerase that we're not going to disrupt that transcription process. But what it does mean is that 3 and 4 then cannot form a hairpin with each other. That means that you're going to continue with translation as usual to make more tryptophan. Another overview thing, in case that's helpful. Any questions about the tryptophan operon? Cool. Okay. So regulons we're sort of zooming out even more. Regulons are a group of operons, and these operons each code for functionally related proteins. It's sort of like an operon of operons, which is weird. This regulon is going to, all the genes in this pathway are controlled by a common regulator, and that's going to allow us to have a coordinated response across all of these operons. So the example that Professor Holmes gave in class was that if you are in a situation where you have low glucose in your cell, again, you're going to have a lot of this secondary messenger, cyclic AMP. And that means that you want to activate all possible pathways for breaking down sugars. So instead of just activating the lactose sequence, the lactose operon, the lac operon, you're going to activate all of these different operons which break down different kinds of sugars. That means that if the body is in a situation with low glucose, you can hopefully break those down quickly and regain that energy charge that the body is missing. Another example of a regulon and an example of a repressed state that we mentioned earlier is our mechanism for DNA damage repair. So again, this system is repressed. It's off until we need it. And there's 2 main proteins that we're going to talk about. The first one is LEX-A. So LEX-A, it's encoded by this gene right here, and it's these sort of orange circles in this diagram. LEX-A is going to act as a repressor. It's going to repress all of these genes that encode proteins used for DNA damage repair. And the second protein that we're going to talk about is REC-A. So REC-A, on the other side here, it's these sort of green circles, is the protein that recognizes DNA damage. So if you're looking down here, if you have a situation where your DNA is damaged, what's going to happen is those REC-A proteins from the cytosol, from the cytoplasm, are going to go and bind to that DNA. That means you're going to have a lower concentration of REC-A in the cytosol, and you're going to make a bunch more of it. It's sort of similar to like Le Chatelier's principle from Gen Chem, like an equilibrium thing. What that reaction does is it causes the proteolysis of all of your LEX-A proteins in the cytosol. So if you have less LEX-A in the cytosol, the same thing's going to happen, where the LEX-A that was bound to all of your genes across here is going to come off of the genes. It's going to get cleaved, broken up. But what that means is that all of these genes are no longer repressed. You can transcribe and translate them as needed, and you can use those proteins for DNA damage repair. So we talked a lot about transcriptional control. We're going to talk a little bit about controlling translation, which is a bit different, but there are several ways to do that as well. So the first one that was mentioned in lecture is controlling ribosome synthesis. So as protein demand increases, you're going to need more ribosomes to make those proteins. And the way that we regulate that is taking note of the fact that ribosomes are made both of ribosomal RNA and ribosomal proteins. So when we need to regulate ribosome production, we need to balance that transcription and that translation. So on each ribosomal operon, there is one ribosomal protein, as you can see in this diagram here. And that ribosomal protein can act as a translational repressor. The way that that's going to happen is there's two different states. So if you're in a state where ribosomal RNA, you have more ribosomal RNA than you do ribosomal protein, what's going to happen is that ribosomal protein will seek out and find that ribosomal RNA, and you're going to form ribosomes as you need them. Ribosomal proteins have a high affinity for ribosomal RNA. So if that's the state that you're in, that's what's going to happen. On the other hand, though, if you have more ribosomal protein than you do ribosomal RNA, you have excess proteins, they're in abundance, and what's going to happen is they're going to bind to their own mRNA, and they're going to inhibit their own translation. So when you have excess ribosomal proteins, you're going to stop making ribosomal proteins. When you don't have enough, you're going to keep making them. Another example of feedback inhibition, like the ones we've seen in the class earlier. Another example of a way that we control translation is with the stringent response. So this is what happens when an uncharged tRNA is going to enter that A site. It's sort of like what's happening in the tryptophan operon, regulating tryptophan with the attenuator. But when you have an uncharged tRNA that enters the A site, that means that you have a low amount of amino acids in the cell. You're unable to charge that tRNA. And that's going to cause translation to slow down. The body's going to recognize that translation is slowed down, and this protein called REL-A is going to bind to the small subunit of the ribosome, and it's going to do this reaction where we're adding a phosphate group to GTP, making PPGPP, which is also known as stringent factor. Stringent factor is a second messenger. So what stringent factor is going to do is it's going to not only upregulate the translation of mRNA that encodes proteins used in amino acid biosynthesis, but it's going to also downregulate all other processes in the central dogma. So what that means is that if we have low amino acids, we're going to focus all of our efforts towards making machinery that makes more amino acids, and we're going to forget about doing other things in the central dogma, because you can't do much without amino acids in the first place. Any questions about that? Okay. Another kind of translational control is RNA-based translational controls, and there's two different mechanisms that this happens by. The first one is trans, which means different RNAs are going to bind to specific mRNAs. That's a bit confusing, but it just means that mRNAs are not going to be regulating themselves. The example given in lecture is RPOS. So RPOS is this mRNA used here. And when it's by itself, it forms this hairpin loop, which of course means that you can't translate it, which is a problem. So the body solution for this is using these two proteins, HFQ and DSRA. So HFQ is less important for this example, but what it's basically doing is sort of turning this way, sort of like an ATP synthase type thing, and it's going to unwind that DNA, unwind the RPOS to sort of pull apart that hairpin. And then DSRA, which is our RNA, is going to bind there and maintain that structure. So in this case, DSRA is helping us to maintain the structure of RPOS so that RPOS can be properly translated. The other kind is cis translational controls. And in that situation, you have an RNA strand that is folding into what's called a riboswitch. And in that state, it can be sort of turned on and off. Yes, you also call these aptamers. Riboswitches are aptamers, which means they just have a specific pocket where specific ligands can bind. And as ligands bind, what's going to happen is these riboswitches are going to change their state, they're going to change their structure, and turn off or turn on protein synthesis as needed. In the example from lecture, we have iron 2+ ions as our ligand, and they're going to bind into that binding pocket of the aptamer of our cis RNA. And that's going to cause a conformational change that increases protein synthesis, likely for something related to processing iron in the body. If you have a lot of iron, you need a way to process it. Okay, recombination. So we discussed this in the context of prokaryotes, but Professor Holmes also mentioned that yeast can do something similar. So important to note that both prokaryotes and eukaryotes can do this. There will definitely be questions on the test related to distinguishing pro from eukaryotes, so I would look at the colors in this slide for review, or anything else that you've been doing. So what's happening in recombination, in the example that Professor Holmes gave in lecture, is this version of Salmonella has a flagella, it has this tail that is made up of only one protein at a given time. That's either FLJB or FLIC, and it cannot be a combination of those two. It's just one or the other. So what happens after about 1,000 generations is the flagella switches proteins. It becomes made of a different protein, which means that immune systems have trouble recognizing and detecting and cleaving it, stopping it from doing whatever it does. So the way that this works genetically is that you have this sort of promoter that can change directions. So in this case, your promoter is facing to the right, which means that you are going to transcribe this gene for FLJB. That's one of our two kinds of proteins that make up the tail. You're also going to produce FLJA, which is a protein that represses our other pathway, FLIC, as a small note. And you're also making Hin recombinase. Hin recombinase is a protein that can recombine DNA so that the promoter is facing the other direction. You're going to sort of flip this promoter. Before it was facing the right, now it's facing the left. And then you're going to transcribe and translate genes related to the formation of FLIC. And that allows the Salmonella to change the protein that makes up its tail. Any questions about that, about recombination? Okay. So we talked a lot about prokaryotic regulation. We're going to talk a little bit more about regulation of eukaryotic gene expression. A lot of these things are things that we have mentioned before. I know Alex mentioned chromatin remodeling, euchromatin and heterochromatin, but as a quick review, euchromatin is loosely packed. When you're in this state, it allows for gene expression, and heterochromatin is this sort of tightly packed situation here, which means that transcription machinery cannot get in there, and as a result, you have no gene expression. You can control this by acetylating histone tails. When histone tails are unmodified, their positive charge is going to attract DNA and wrap tightly, which is going to form heterochromatin. On the other hand, when you acetylate these histone tails, the charge is going to be neutralized. Packing is loosened, and you have euchromatin. Removing these acetyl groups is done by histone deacetylases, and adding acetyl groups is done by histone acetyltransferases. Just a couple enzymes to keep in mind when you're studying this pathway. Another way that we control eukaryotic gene expression is with managing this sort of location of transcription factors, sites where they bind, things like that. So as we mentioned earlier, control elements in eukaryotes can both be proximal and they can be distal. In this case, distal means just in general further away. Some examples that Professor Holmes gave in class is it can be upstream of your open reading frame, downstream of it. It can be within introns or even on other chromosomes. So there's a couple ways that we use that difference to help transcribe and translate eukaryotic genes. We have these things called architectural regulators. That's these here, these HMG proteins in this case. And what that's doing is sort of twisting the DNA so that they're close together in 3D space. That's bringing our transcription factors closer together to allow them to have an impact on the situation. You also have this mediator protein or co-activators. These are proteins that sort of hold that 3D structure together, again allowing the transcription factors to be closer in space. So the way that we regulate eukaryotic gene expression has a lot more to do with orienting the DNA itself, whereas in prokaryotes, it was pretty straightforward. You either had an operator sequence or an activator binding site. Any questions about that? Okay, cool. Professor Holmes also mentioned yeast gal. Gal stands for galactose. This is sort of a comparison to the lac operon. Of course, remember that eukaryotes don't have operons, so we have another way to induce the breakdown of these sugars as we need. The example that he mentioned are these proteins GAL80 and GAL4, which are going to bind to all the GAL genes at upstream regulatory sequences. And when that happens, they're going to trigger, if possible, the binding of GAL3 to galactose. When GAL3 is bound to galactose, this is sort of acting as another molecular sensor, sensing our levels of galactose in the cell. When that happens, when you form that compound, that's going to activate the process. You're then going to cause GAL gene-encoded proteins to be made, and you can process galactose. You definitely don't need to know this process in a lot of depth, but just, of course, remember, again, that eukaryotes don't have operons, and that we have a sort of separate way of managing 3D space and breaking down sugars, encoding the genes that break down sugars. Okay, one of the last examples of eukaryotic gene regulation is intracellular signaling. This is something you might have seen in something like physio, related to hormones, even earlier in this class. But there are two kinds of receptors that we are going to mention in this situation. The first one is type 1. These are receptors that are hanging out in the cytoplasm, which means that our hormone, our signaling molecule, is just going to diffuse across the plasma membrane. It's going to bind to the receptor in the cytoplasm. And that hormone receptor complex are then going to cross this membrane into the nucleus, where they're going to regulate translation. The other kind, type 2, you have your complex, your receptor, already in the nucleus. These are often repressors. So in this case, for example, we have a repressor hanging out on our gene. Our hormone this time is going to diffuse all the way through the plasma membrane, through the nucleus membrane, and onto the gene. It's then going to disable that repressor so that we can activate the gene. So the only difference in this case is where the repressor is hanging out, whether in the cytoplasm or in the nucleus attached to the DNA itself. The examples he mentioned in class is that type 1 hormones are generally steroid hormones. Type 2, an example, is thyroid hormones. That's not something you need to know, but in case you're interested. Okay. Controlling translation in eukaryotes is, of course, a lot different from the way that we do it in prokaryotes. There's sort of three different things that we're going to mention in this case. Again, this is sort of a review from the previous lectures, but eukaryotic mRNA has this 3' untranslated region, or UTR. That's this section over here. And in that area, proteins can bind and trigger changes that inhibit translation. That's what these repressors are doing over here. Another thing we can do is that eukaryotic translation involves these eukaryotic initiation factors, which we can phosphorylate, and that's going to inhibit translation. And the last example Alex mentioned are microRNAs, which are going to bind to specific sequences, and when they do so, they're going to block the ribosome from translocating, and they're going to inhibit translation. Okay, any questions about lecture 22, gene regulation? Awesome. One more. DNA technology. Okay. So Professor Holmes started this lecture by mentioning how we began isolating DNA. The way that this person did it, Chargaff, I don't know how to say that. Chargaff? Thank you, Alex. He took a bunch of species, isolated their DNA, and he found that A and T and C and G are always in 1-to-1 ratios. That sort of gave us a preliminary idea of what DNA structure could be like, or what some components could be. We can now back this up with our understanding of purines and pyrimidines, how A will always bind to T, and C will always bind to G. You can quantify the amount of nucleotide that you have with UV spec. Specifically, at 260 nanometers of light, different nucleotides are going to have different values of epsilon in Beer's law. Yeah, just a note about how those are identified. A lot of the techniques that we're going to talk about today involve isolating strands of DNA. So the easiest way to do that is you can disrupt the hydrogen bonds between the two strands, those base pairing interactions, by heating up the strands. You can use a bunch of different agents. But Doing so is going to cause the strands to denature, which just means they are separating from each other. This isn't like a permanent solution. When these are cooled again, if you use heat to separate them, the strands are going to reform those hydrogen bonds and going to reform those base pairs. That process is called annealing. So denaturing and annealing are opposites for the sake of this unit. Different sequences of DNA are going to require different temperatures to denature them. And that's because of differences in amount of AT and GC. Again, remember from lecture 18 that AT is going to form 2 hydrogen bonds and GC is going to form 3 hydrogen bonds. So breaking GC is going to require more energy. If you're using heat, it's going to require more heat. So the way that we can quantify this is with a melting curve, and we can use these curves to sort of determine loosely AT versus GC content across genes. So a gene with higher GC content is going to require more heat to denature, and its curve is going to be shifted further to the right. The specific value that we use to measure this is Tm. That's the melting temperature at which half the DNA strands are separated. Again, I wouldn't be surprised if there's like a multiple choice question that's giving you 2 of these curves, telling you which one has a higher GC content and how do you know, or something like that. Okay, recombinant DNA is also a technique that we're going to use in a lot of these other techniques that we discuss. So recombinant DNA is these artificial plasmid structures that we are forming by combining DNA across a bunch of different organisms. Plasmids, if you remember, are found naturally only in prokaryotes, but we are going to be taking them and adding in prokaryotic genes, eukaryotic genes, whatever we are interested in. If we add a eukaryotic gene to this system, to this vector, this plasmid, we can then put it back into the prokaryotic system, and the prokaryotic system will will transcribe and translate that eukaryotic gene. The easiest way that we can combine these genes together is using restriction endonucleases, which we will talk about in a second. Okay, so what restriction endonucleases do is they are binding and cutting the DNA at extremely specific sequences, known sequences. These are usually going to happen at palindromic sequences. These are sequences that if you look at them, 5' to 3', they are the identical. Or another way to think of that is that they're complementary to themselves. When restriction endonucleases cleave, they can either leave sticky ends, that's these here, these overhangs, sort of L shape, or they can leave blunt ends, which is no overhang, you just have these sort of straight edges. There's a lot of things that we can do to manipulate these plasmids, use them to encode the gene that we have of interest. So what you can do is use that restriction endonuclease, a specific one, and cut both the plasmid and your sequence of interest in specific places, and then reattach the gene back into your cloning vector. Again, you can then put that back into bacteria for gene expression. A problem, though, is that the problem that we want to solve is that how do we know that this worked? How do you know that the bacteria took up the plasmid and that cloning and replication was successful? And the way that you can do that is in the gene that you are inserting into your cloning vector, you can include a gene that encodes for something that creates antibiotic resistance in the bacteria. Then what you can do is you can treat that bacteria with your antibiotic, and any bacteria that stay alive are the ones that successfully took up your plasmid, and any that die, of course, didn't have that gene, so they were unable to survive. That's just a way to sort of separate out the bacteria that have successfully taken that cloning vector. Okay. A pretty common experimental technique and the basis for a couple other things. We're going to talk about qRT-PCR later, but this is the sort of first step of that, polymerase chain reaction, or PCR. What we're going to be doing is we're going to amplify one specific region of the genome, and we're going to ignore other interests. So if you want a lot of a specific sequence of DNA, you can use this technique. This is going to happen in a couple steps. The first thing that you're going to do is you're going to heat up the DNA strands. That's going to separate them. That's what we talked about earlier. The denaturation process. The next thing that you're going to do is you're going to design these specific primers that are going to anneal to the region of interest, the region that you want to clone to make a bunch of copies of. You can add sort of just this end, this 5' end, but you can also add additional information onto your primers. For example, if you add a restriction endonuclease site, you can then really easily use a restriction endonuclease to cleave it and insert that gene into a plasmid of interest. You're then going to cool the strands very slightly just to allow the primers to anneal, and then you're going to use DNA polymerase to build these proteins into full strands of DNA. It's important to note, though, that you can't just use normal DNA polymerase. You need a polymerase that survives at extreme temperatures because we're doing so much heating and cooling. And we're going to be using Taq polymerase. Taq polymerase is taken from thermophilic organisms, which just means organisms that survive at extreme temperatures. And it's going to be used in this process as we heat and cool. You're then just going to do that a bunch of times until you have amplified the gene to the extent that you need. And now you have a bunch of copies of your gene of interest. Another technique is gel electrophoresis. This is sort of the DNA-RNA equivalent of SDS-PAGE, which we talked about in lecture 3. But what it's going to do is we're going to separate DNA or RNA by size and charge. So what's going to happen is we're going to run this gel. We're going to have an electrode with a positive charge down away from our wells and a negative charge up behind our wells. And what that's going to do is, because DNA has a negatively charged backbone, the DNA is going to migrate down the gel. Smaller proteins are going to migrate further. Larger proteins are going to travel less distance. In order to visualize this DNA, you're going to use something like ethidium bromide. Ethidium bromide is a dye that fits into the gaps of DNA and makes it fluoresce, just so it can be easily seen. So when we want specific DNA, a problem that you could run into is that DNA, of course, contains introns. So if we just want the DNA of a specific protein, if we just want the exons, we are going to create cDNA. cDNA stands for complementary DNA, and we're going to use reverse transcriptases to make this DNA from RNA. So the basic steps of this are, we are going to take the cells and we're going to lyse them. We're going to purify their mRNA, or whatever mRNA we're interested in reverse transcribing. These mRNAs, especially if they're from eukaryotes, are going to have a poly-A tail, so you can attach a poly-T primer in the direction that you need. If it's not eukaryotic cells that you want cDNA from, you just need to know some sequence of it so that you can form that primer. When you have the primer and the primer anneals, you can use reverse transcriptase to reconstruct DNA from that mRNA. And then you can degrade the RNA backbone with, you don't have to know this enzyme name, but RNase H. You're just sort of getting rid of that extra mRNA so that you have room to use DNA polymerase to synthesize the complementary strand. So now you have double-stranded DNA that has only exons. It has only the information of your proteins of interest. So we talked about how you can sort of ensure that bacteria took up the gene of interest using antibiotic-resistant genes, but you can also control the expression of the genes itself. The way that you can do that is you can add an inducible promoter to your plasmid and make that inducible promoter easily expressible in a way that you can control experimentally. An example of that is you can add a sequence that has a repressor. You can then add that repressor, which hopefully can be easily removed. So when you want to transcribe that gene, you can add whatever removes the repressor that you added the code for, and the gene will be expressed in cells. Another means of purification is epitope tags. So epitope tags are just a way of adding another protein to your protein of interest in a way that you can filter them out. The way that you do this is you take the gene for your epitope tag and you add it immediately next to your cDNA without any stop codons. Because you want, when translation occurs, you want to produce your protein of interest and you want to have it attached to that epitope tag. An example of an epitope tag is GST. So, as we mentioned before, you're going to add the coding sequence of GST to the plasmid immediately next to your protein of interest. As a result, that plasmid, when it's translated, is then going to form protein of interest with a GST tag attached to it. And now you can easily purify that protein because you know properties of GST. GST is going to bind to glutathione. So you can use a column, again, column chromatography from lecture 3. You can use a column to separate out proteins with GST attached. Then, once you've done that, you can wash out those proteins with a solution of glutathione or a solution of GST, and you have separated out your protein. Another example of an epitope tag is GFP. GFP isn't necessarily used for separation of these proteins, but it can be used to easily visualize them, quantify the amount that there is. GFP is just a protein that emits light when it's excited. So as you did before, you're going to put the genetic code for GFP right next to your protein of interest. When it's translated, you're going to have your protein of interest with GFP fused onto it, and then you can easily visualize it. This also allows you to track different proteins in the cell. GFP now comes in a bunch of different colors, so you can give one protein one color, another protein in another color, and see how they interact or where they go. Any questions about any of the techniques so far? Okay. The example that Professor Holmes gave of recombinant proteins is insulin. So insulin was discovered as a cure for diabetes and then was the first protein that was ever sequenced. It later became the first protein that was artificially synthesized. And we were able to purify insulin from cows and from pigs and sell it for diabetes treatment. But the easier way to produce insulin is to use recombinant DNA. So you put the gene that codes for insulin into a cloning vector. You put that cloning vector, that plasmid, into bacteria. And then it's just going to go crazy and make a bunch of insulin. Okay. So if you want a way to manipulate a specific part of the sequence, the example Professor Holmes gave is if you have a codon like CAG and you want CG, for example, you're going to use site-directed mutagenesis. This is sort of an old technique. We'll talk about better ways to do this, but this is sort of the simplest way to do that. If you want to change the sequence, manipulate it at a specific site, this is what you'll do. The first thing that you do is, as before, you're separating the two strands of DNA. You're separating out that plasmid. And then you're going to anneal primers with that mutation that you're interested in. So if you want CAG to CCG, you'll add that primer there. You're then going to use DNA polymerase to complete both of the strands. Again, this is happening separately. The left strand and the right strand, you're using different primers, and DNA polymerase is acting separately on each of them. And now what you have, once you break down the parental DNA, if you remember from last unit, parental DNA is methylated. It has a CH3 attached. So you can use nucleases specific to methylated DNA to break down your parent DNA. And then you combine those two genes together, and you have a plasmid that just has your that mutated region, the region that you changed. We have a new way to do that that is a lot easier and a lot more specific. The problems with site-directed mutagenesis in plasmids is that, of course, you have to do it in plasmids. If it's some structure that can't function in a bacterial cell, or for whatever reason can't survive in a plasmid, you are unable to change the sequence. So that's what CRISPR-Cas9 does. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It was discovered as the sort of immune system of bacteria. And all this is, as you can see in this picture, is you have these palindromic sequences spacing out chunks of virus DNA. So what's happening is that when a virus infects bacteria, the bacteria is going to destroy the virus and save some of its DNA. It's just going to store it in that chunk that we described earlier with palindromic sequences in between. And then when the virus comes back, the cell is going to recognize that viral DNA. It's going to use that DNA that we stored earlier it's going to transcribe it and make guide RNA. Guide RNA can then be used to bind to the original DNA sequence. So you can take that guide RNA and it'll take you to the virus, which you can then use to destroy the virus. Bacterial cells don't have a specific way of destroying the viral genome, or they do, but they don't have a specific way of modifying that individual gene, so that's where Cas9 comes in. Cas9 is CRISPR-associated nucleus. It uses that guide RNA made by CRISPR to find a specific sequence of DNA, which it can then cleave or manipulate as necessary. So experimentally, what we do with CRISPR-Cas9 is you design the guide RNA yourself. The guide RNA is based on whatever gene you want to target, whatever sequence you want to target, and that's going to show the CRISPR-Cas9 complex where to cut. There's a bunch of different things you can do with CRISPR. Super cool. You can cut 2 strands. You can add sequences directly into the genome. Of course, you can remove sequences of interest. And you can also fuse Cas9 to a repressor to find a specific sequence and then tightly regulate gene expression. So Cas9 is a great way, CRISPR-Cas9 is a great way to find a specific sequence, manipulate it, add it, remove it. And it's a lot more site-directed than something like site-directed mutagenesis. Any questions about CRISPR? Cool. The example that Professor Holmes gave in lecture of CRISPR-Cas9 is using it to treat sickle cell anemia. So what you can do is you can isolate a patient's stem cells, and then you can use CRISPR to reprogram them. There's 2 different ways to do that. You can either repair the beta globin gene, that's the mutated gene that makes a part of hemoglobin. You can repair it so that the correct hemoglobin is made, or you can also modify promoter sequences so that fetal hemoglobin is made, and that's just sort of non-impaired hemoglobin. Once you've reprogrammed those stem cells with either method, you can then take them and put them back into the patient's bone marrow. The body will make a bunch of copies of it, and in turn will make the correct hemoglobin molecules. Super cool. Okay. We're going to talk about DNA sequencing. There's a lot of ways to do this. This is probably the easiest, but it's also probably the slowest. Sanger sequencing uses what is called dideoxynucleotides, or DDNTPs. These are just nucleotides that lack both the 3' OH group and the 2' OH group. What that means is that you can't extend a strand of DNA that has one of them, because you need that free OH group normally to extend your DNA strand. So when you introduce a DDNTP, what's going to happen is that replication is going to terminate, which is why we call this terminal sequencing. So what you do is you take one of your dideoxynucleotides, for example, DDATP, you put it in a well with a bunch of normal TTP, CTP, and GTP, and what's going to happen is you're going to form a bunch of these strands. You're going to form strands that are cut off where all of the adenines in the sequence are, which then, if you accurately keep track of which the lengths of these strands, you can use to sequence. So in this case, we're starting with a G, an A, a T, a T, a C, and so on. Again, wouldn't be surprised by a test question that was something like looking at this Sanger sequencing electrophoresis gel, sequence the DNA strand in question. Again, this is sort of the most bare bones way to do this. One of the newer ways to do this is to just use fluorescence and then automate it. So what you're doing is you are giving each of your dideoxynucleotides a different color using a fluorescent tag. Again, one color for each of the bases. And then you can automatically sequence DNA by doing what you did before, that sort of Sanger sequencing where you put some of each of your DDNTPs in each well. And then, yeah, you can use tech to analyze the wavelength of each of these nucleotides based on whatever color you made each of them, and learn the sequence of it. There's some problems with this, though, which is that you need very, very pure DNA to be able to make it fluoresce without making anything else fluoresce. And you also need to know some part of the DNA sequence to begin this process. You need primers to do this. The solution to this, which is even newer, is next-gen sequencing. We're doing this by synthesis, so it is sometimes called sequencing by synthesis. With this technique, you don't need to know anything about the DNA because you don't need specific primers. Well, rather, you are making artificial primers, which you then add via ligation to your fragments of DNA. But all you're doing is you're taking a DNA sample, a long gene, and you're chopping it up into random fragments using a restriction enzyme. You're then going to take these fragments, affix them to known adapters, which are going to act as your primers. These are known sequences. And you attach this to a flow cell. Professor Holmes showed a picture of that. It's just like this little chip-like thing with little strands. And you're going to keep doing DNA replication. After each round of replication, you are taking a picture, and you can see how the colors changed at each strand, which will tell you which nucleotide you added. So these pictures are sort of an example of that. If you follow this, for example, if yellow is A, whatever, blue is T, red is C, you have A, T, C, G, G, and so on. You can then use computational analysis to do that for each of the strand. Of course, you chop this up into a bunch of fragments, so you have to do it for a lot of fragments at a pixel by pixel level. And then you have the sequences of a bunch of random strands of your DNA. You can use more computational analysis to match these sequences together, see where they overlap, and form your whole gene sequence. Okay, we're almost there. We talked about gene sequencing. We are going to talk a little bit about measuring gene expression. So the way that we do this is with QRT-PCR. Most of the steps of QRT-PCR are identical to the steps of normal PCR. So I didn't include sort of a step-by-step of that. The only difference, though, is that you're going to use fluorescent probes. These fluorescent probes are these sort of hairpin structures here that has both a fluorophore and a quenching molecule, a quencher. The fluorophore is a molecule that fluoresces. The quencher is a molecule that quenches light. It absorbs light. So what happens is that this hairpin structure, when the probe is bound to a gene, is going to stretch out, and it's going to separate the fluorophore from the quencher. When these are separate, you're going to emit light because the quencher isn't close enough to quench that light. And when the probe is not bound to the gene, it's in this state where the two are super close to each other, so no light is going to be emitted. You don't have any glow. The way that you analyze QRT-PCR data is you get these sort of curves, and you are looking specifically at the cycle threshold value. Cycle threshold might be something you decide before your experiment. It might be something from literature, but it's just a sort of baseline level for signal that you are looking at. And if you have a lower cycle threshold, again, that means you're crossing at an earlier cycle. That means that the gene is expressed, there's greater gene expression of that specific gene. The reason for that is that if you have a lot of the gene, that means you're going to have your fluorescent probe in this stretched out state a lot more than in this state, which means you're going to be emitting more light. So that's sort of a way to quantify gene expression relative to other genes or just by itself. Okay, one of the last techniques we're going to talk about are microarrays. So you can use microarrays to compare levels of gene expression in two different conditions of a cell. So that could be like a healthy cell and a pathological cell, wild type versus modified. The way that you're going to do this is you're going to purify all the RNA out of both of these samples, and you're going to label each of them with a different color. For example, in this picture, we have normal RNA labeled with green, and your pathological cell RNA labeled with red. You're then going to hybridize these to known sequences on a chip. The chip's going to have many wells of known DNA sequences. You're going to transcribe these, reverse transcribe the mRNA to form DNA. And the appearance of, I don't know why that says used. The appearance of colors such as red or green in this example is going to indicate predominance of expression in one state. So if you have a lot of red, that means you have a lot more expression of that gene in pathological cells as compared to normal cells. If you have green, you have a lot more expression of that gene in normal cells. And if you have that in-between color, in this case that yellow, that means you have pretty equal expression across both of these. This is another way to use light to quantify gene expression, but you're using this specifically across two different states. I think it's useful to know when you're going to use these techniques, what each of them does as well, because there could definitely be a test question that's like, hey, you have a cell in a healthy state and a disease state. How do you determine levels of gene expression? Okay, that's all our techniques. Last slide. There isn't a lot of testable material here, but just important to note that the majority of the human genome doesn't actually code for proteins. It's got introns, we've got lines and signs, which we didn't cover in this class. You don't need to, of course, measure the numbers of these. Just know that the majority of your genome contains non-protein coding genes. Genes also have polymorphisms. These are small variations in the gene sequences that are going to cause variations in gene phenotypes. This causes diversity in phenotypic traits. And you also have these things called single nucleotide polymorphisms. These are variations in a single nucleotide within a sequence, and these often occur within a specific population. You can identify a specific individual by their single nucleotide polymorphisms, which is what we use to, for example, identify criminals at a crime scene. Lastly, haplotypes are groupings of these single nucleotide polymorphisms that can be used to make more accurate phylogenetic trees. This is what something like 23andMe uses to trace ancestry. Okay, that's everything. Thanks, y'all. Yay!
Like studying, just like a little tip, be comfortable with reading like gel electrophoresis, especially they could give you, for example, like a couple of wells susceptible to different enzymes, and you get different number of bands corresponding to each well, and then they'll basically give you circular plasmid and they say, where does it cut the enzymes? Not that you need to know specifically how far the base pairs are, but just know where they cut, if they cut twice or cut only once. That's something to know as well.
Yeah. If you have specific questions, feel free to come up to us, post on Ed. I'll be there tonight and tomorrow. Best of luck on the exam. Y'all got this. Last one.
We're done.
With the plasma thing you were saying,
Yeah.
genetics, they code the plasmid.
It'll be very similar to that, but much more simplified. Like, it's not going to be a headache at all. You'll basically get like a control, the DPNN, digest, whatever it is.
Probably the worst thing I did in genetics.
Yeah, no, it'll be a lot more simplified. Yeah.